A very good morning to all. Welcome to the final day of GYSS. My name is Wei Jing from the National Research Foundation. A few housekeeping announcements before we kickstart today's program. Do put your mobile devices to silent so that the program can proceed uninterrupted. We would also like to hear from you regarding your experience at GYSS these few days. You have received the feedback form that we have sent to you yesterday, so we'd like to seek your assistance to fill up the feedback form. Next, do follow us on social media for updates on GYSS if you have yet to do so. You can find us by searching for gyss.sg on Facebook and gyss_SG on Instagram and Twitter. And do also join us in the action by tagging us and using the hashtags. So that's all for the housekeeping announcements. Thank you for being with us here again. I hope you have had the opportunity to explore Singapore, whether on your own or via the social programs yesterday. But if you have not, we have lined up a closing event at Sentosa Island for you later this evening. But to get us there, let us now start with the first segment, which is the Young Scientist presentations. We invite Prof Leo Xiaogang from the Singapore University of Technology and Design to chair the session, and our presenter, Awais Kasim. Regrettably, another of our young scientists, Dr. Rachel Lapan, is unable to join us in person today, but she has kindly prepared a video recording. So I'll now hand over the session to Prof Liu, please.
Okay, ladies and gentlemen, a very good morning. My name is Xiaogang. I'm from Singapore University of Technology and Design. I'm working on chemistry research, specifically computational chemistry. And today it's my honor to be the chair for this morning session. So welcome to Singapore, welcome to SUTD. I seldom have a conference starting at 8 a.m. It's really early, so you are really the early birds and you are awesome. Okay, so our first speaker will be Dr. Rachel Lapan. Rachel is now working as a research fellow at Monash University in Australia. Her research is about microbiology. Specifically, she's studying how microbial is living in the harsh environment and how they interact with our world. Microbial are very small, but the impact could be big. So let's watch the video from Dr. Lapan.
Okay, hello everyone. My name is Rachel Lapan. I'm a postdoc at Monash University, which is in Melbourne in Australia. And even though I can't be here in person today, I'm recording this from my hotel room. I'm really excited to share with you this new research that I will be embarking on this year about microbial life in the atmosphere. But I do want to begin by talking about microbes in general and also how some of my previous work led me to this really exciting area. So we really are living on a microbial planet. Microbes are essentially the foundation of all ecosystems on Earth because in our guts and in the guts of other animals, they aid in digestion and metabolism. They assist the functioning of our immune system, and they help us to defend ourselves against pathogens. In the world's oceans, microbes are making up most of the biomass there, and they play important roles in decomposition and photosynthesis, so really forming an important part of the carbon cycle. And they have similarly important roles in soil, particularly where they form close relationships with plants. Microbes are also inherently connected to health. Not only human health, but also animal and environmental health. So these three areas shown down on the bottom left are really linked and dependent on each other, which is a concept referred to as One Health, the idea that you need to treat issues in these areas in a holistic way. And some of the earlier work that I did as a postdoc focused on pathogen surveillance in this project called RISE, which is still ongoing. And so here we were aiming to improve water and sanitation in informal settlements with some new infrastructure. And I was involved in developing techniques to keep track of the many different diarrheal pathogens across humans and animals and various aspects of the environment. So doing this work really sort of, I guess, opened my eyes and taught me about the connections between human and animal and environmental health, this One Health idea. But something that I find really exciting about microbiology is how microbes can live in these very unique places where nothing else can. They can live at extremely hot temperatures, in hot springs. They can be in very acidic conditions. And also in the cold and dry valleys of Antarctica, there are microbes living where you would think that life couldn't be. And so this interest really brought me to the field of microbial ecology, which looks at the microbial communities that live in a certain place, how they interact with each other, and also how they interact and are really shaped by their environment. So when it comes to studying microbial ecology, we need to do things fairly differently to traditional microbiology, because a lot of microbiology is based on culture-dependent work, which means that we have to grow the microbes in the lab before we can study them. However, the majority of the microbes out in the environment, in most places, in fact, and especially in these more unusual places on the planet, are uncultivated, which means that we just haven't yet worked out how to get them to grow in the lab, and they may not like to grow that way. So especially when we're looking at an entire community of microbes, and there might be hundreds of species there, this is where metagenomics comes in. So we've talked about DNA sequencing so far in this conference. So instead of growing the microbes with metagenomics, we're looking at their DNA. And metagenomics takes all of the DNA from a sample like soil or water, and we sequence everything so that you can look at the genetic information of every organism that is in there, all of the microbes and anything else. And so this tells us two very important things. It tells us firstly about taxonomy, or the profile of who is there in the community and how abundant they are compared to other species. And it also tells us about their function, because in doing all of this DNA sequencing, we can look at the genes that they have, and that tells us about the sorts of things that they are capable of doing. And so what I want to say here is that my real sort of passion now is doing work to understand how microbes can survive in challenging environments. The idea that, you know, life is sort of living in places where it really shouldn't be, but it finds a way nonetheless. And the reason why I find this so exciting is because microbes are extremely metabolically flexible. And what I mean by this is that generally we eat carbohydrates and we respire oxygen. We sort of don't have any other options. We can't really live any other way, and we would die without either of those things. But so we're not very flexible. But microbes can eat all sorts of organic and inorganic compounds to get their energy, including atmospheric gases. And they can also respire oxygen like we do, or they can live without it, live anaerobically and respire something else like nitrate or sulfate instead. And so different organisms can use different strategies for survival, but it's also quite common for any given organism to be able to use multiple survival strategies depending on what is available in their environment. So they're very flexible, they're adaptable. And so I'm really fascinated by this, and particularly, as I mentioned, the ability for microbes to use atmospheric trace gases as their source of energy. And this is the focus of the lab group that I work in, and there's been some really exciting advances here in recent years. So I'm going to talk about that a little bit. So as you will know, the atmosphere is mostly nitrogen and oxygen, which does a great job at supporting aerobic life. But there's also a lot of other gases in the atmosphere that are present in trace amounts. And these gases, particularly hydrogen, carbon monoxide, and methane, have recently emerged as this sort of hidden energy source for microbes. It's only recently that it's become appreciated just how widespread and important these gases, or widespread the ability to use them, and how important the gases are, these trace amounts in the atmosphere, in sustaining a lot of different microbial communities. And much of the work that our lab has done has focused on how microbes can use these gases, particularly those three that I mentioned, as their source of energy. And so one of the places where this is really important is in Antarctica. So photosynthesis, which would usually be the main call it primary production, you know, where most of the biomass comes from, it's really challenging to do photosynthesis in Antarctica because it's a process that requires water, and Antarctica is actually very dry. For parts of the year, there is also no sunlight. And the soils in these Antarctic deserts are actually quite nutrient limited in general. And so we've found that there's actually this huge diversity of microbes. And in fact, the majority of the community, you can see the trace gas oxidizer abundance down there, is actually capable of using the small amounts of hydrogen and carbon monoxide from the atmosphere to sustain themselves. So many of the members of these microbial communities are essentially living on thin air. And so in the greening lab, where I work as a postdoc, I have worked across a number of different projects looking at how microbes can live on air in different places. So one of these was in desert soils. So I looked in hot and cold deserts, and we found that hydrogen is a really important energy source in the desert. We've done these experiments that you can see where we sort of very sensitively measure the amount of atmospheric hydrogen that is consumed over time by the microbes in these soils. I've also worked on a project in termite mounds where termites actually, they produce a lot of methane and also hydrogen. And we found that the microbes within the termite mound were actually consuming some of these gases released by the termites. And so to some extent, the microbes are mitigating the emissions of methane and certainly of hydrogen. There's a net hydrogen uptake by the mound. And that's how the termites are sort of feeding the microbes in these mounds and this interesting relationship between them. And I've also worked in the marine space where we found that even in the ocean, dissolved carbon monoxide and hydrogen are actually a really important energy source for microbes in the ocean as well. And the microbes that have the ability to use these gases for energy become more abundant in the ocean the deeper you go. You can see that in the graph for both carbon monoxide and hydrogen. And so in each of these projects, it was really the metagenomic approach that allowed us to explore these microbial survival strategies. And so that brings me to how these sort of seemingly very different projects have kind of come together. And, you know, it's come from me doing this work and being around other research going on in the lab about extreme environments and thinking about this, you know, continually wild idea that bacteria can eat gases to get their energy. And then also with me coming from this sort of One Health and disease surveillance kind of background, I was thinking about all of that, and I came across the field of atmospheric microbiology, as in microbes living up in the atmosphere. And I thought, wow, this is really something that's very exciting. It's an area that has not been very well explored at all, at least not in a metagenomics sense, because most of the research about bioaerosols in the atmosphere is about pollen or allergens. There's a lot of papers that are focused on how microbes are dispersed from one place to another by the atmosphere. But there's not that many research groups in the world that are focusing on microbial communities that are actually living in the atmosphere. And so this led me to be thinking, is the atmosphere actually just another extreme environment on the planet that actually hosts this substantial and diverse ecosystem, perhaps, of microbes? And so I took this idea and I applied for and received this early career fellowship that we have in Australia, which really allows me to sort of independently embark on this journey of exploring this research area more, where this combination of my experience and expertise that I built over the past few years is what's really sort of led to my perspectives here. So thinking about the atmosphere as a microbial ecosystem, like I said, there's not a great deal of literature here. The atmosphere has certainly been investigated for microbes, and there is some good work out there, but much of it has really sort of considered microbes to just be transiently moved from one place to another by the atmosphere, and that's it. So this idea that there could be microbes that are calling the atmosphere home, being sort of resilient to the harsh conditions up there, and even perhaps actively doing something in the atmosphere is still quite a very new idea. And I think one of the reasons why this has not been explored very much, or at least not nearly as much as these entire fields of soil microbiology or marine microbiology, is because it's a really challenging thing to study. And so why is that? So firstly, if you want to look at things with metagenomics, it's not something that existed until about 20 years ago. So this sort of world of opportunities that have only recently opened up in order to study this. The other major issue, as you can imagine, is that there's a very low amount of biomass in air. You think about taking an air sample, if you grab a bag of air, there's really essentially nothing in there. So if you're attempting to sort of very sensitively detect microbes and look at their genomes and, look at the entire community that could be up there, it's very hard to do this without contamination from other sources becoming a significant issue. So I have started some work here. Like I said at the beginning, this is a new area of research for me across the next 3 years. This is really what I'm going to be focusing on. But I have made a start, and I'm essentially just taking all of the precautions that I can when I'm sampling. That's a photo of me. So I dress as though I'm working in a clean room when I'm sampling to minimize the amount of cells and dust and stuff that's coming off of me. And then I will be extracting the DNA in this clean room facility that we have at Monash University. Not this one, this one's at NASA, although it might be a bit cheeky for me to say, I'm pretty sure the one that we've got is actually cleaner. So really just taking all of the precautions that I can to prevent contamination from getting into the samples in the first place is really the key, and doing that at every opportunity. So where can I actually go with this research? So obviously the very first thing to do is to really overcome those challenges of maximizing the amount of biomass that we can sample, minimizing contamination. And then given that that's sort of what's almost held up the field this whole time, is once that's overcome, there are so many different ways that all of this can go. So these 3 aims are what I've laid out for my fellowship. I really want to get a thorough understanding of exactly what microbes are up in this microbial ecosystem. And indeed, if it is an ecosystem, I want to understand, secondly, whether they are metabolically active and what they're doing. Are they performing important functions like perhaps regulating the composition of the atmosphere or degrading certain compounds? And then I also want to investigate the role of atmospheric microbes in seeding new environments on land, like in glacier forelands. How do these brand new ecosystems start in an area that was previously barren? So that's what I'm planning to do. But this kind of work really has a lot of connections that I can see, and probably many that I haven't thought of. So firstly, it takes me back to my One Health experience, because the atmosphere is critical in the processes of airborne pathogen transmission, antimicrobial resistance, dissemination of that over potentially large scales, which obviously has health significance. It's really important in better understanding global biodiversity, how the atmospheric microbes interact with those on land and how things change depending on where you are in the world. Regarding the microbial activities in the atmosphere, if you think about it, the atmosphere is covering literally the entire surface of the planet. So it's mass, or maybe not mass, but volume-wise, it's the largest potential microbial ecosystem on the planet. So if there are microbes there that are carrying out important functions like they do in our oceans and in our soils, their impact is probably actually quite critical to the planet. So I really want to look into that. And finally, of course, this work also contributes to our understanding of the limits of life. Just how far can microbes go to survive, both in the context of our planet and potentially on others as well. So I will leave it here by simply summarizing that microbes have and always will shape our planet. They are the dominant life form. We probably wouldn't survive long without them, and they'll be here long after we're gone, I expect. My experience in a couple of seemingly different areas is what led me down this exciting avenue of research, where there is really a lot yet to learn. And once we've overcome those technical challenges, then there are so many avenues for some very exciting research questions yet to come. And I just want to say that interdisciplinary science really is the way forward. Us talking to each other from extremely different fields is how the innovation and everything comes about. So I do welcome your ideas and your questions. I want to acknowledge the funding sources for this work that I'm embarking on, Chris Greening and the Greening Lab. And I'm very sorry that I couldn't be there in person. So please reach out to me on Twitter or by email. I do want to hear your thoughts, your insights, your questions. I am absolutely happy to answer anything. Thank you so much for your attention today.
Although she's not here, please do not hesitate if you want to contact her if you have any questions. And you can see the microbial are small, but they are really tough, living in all kinds of environment. That's definitely something we can learn to cope with challenges in science, right? Okay, our second speaker will be Mr. Awais Qasim. Qasim is from Pakistan. He's currently doing his master's study in the University of Quds Azam University. His research is also about microbiology, and he's dealing with wastewaters, which play a crucial role in our daily life. So without further delay, let's welcome Azam, please.
Hi guys, good morning. And I'm Awais Qasim from Pakistan, and I'm also a Biosecurity Champions Fellow from US, Health Security Partner. Plus, I have certification in Biorisk Management from International Federation of Biosafety Association from Canada. So, I'm the guy who are looking on the positive side of microorganisms, because if you noticed during pandemic or other in life, so people termed microorganisms as germs, and germs as its negative meaning, I can say. So what's pharmaceutical water? Actually, when pharmaceutical contaminants from the industries and leftover products like drugs, it comes to the water, then we can call it pharmaceutical water. So there are contaminant life cycle. There are a lot of sources for pharmaceutical contaminants like agriculture, household, hospitals, and in pharmaceutical industries. And most of the antibiotics come from livestock. Then there are risks for aquatic life. These pharmaceutical contaminants, they can alter the blood profiles. They can cause gender conversion, like estrogen in water. It's evidence that it's converting the gender of the fish. And then the liver dysfunction is there, oxidative stress is there, and hormonal imbalance in aquatic life, mostly in fish, and hyperglycemia, hyperplasia, a type of cancer, and cellular degeneration in fish, immune disruption. And it's actually the difference between alteration in blood profiles and immune disruption here is in immune disruption, some of the cells, they might increase, but in the like in the blood profiles, some of the cells might be increased, but in the immune disruption, like all the white blood cells, immune cells, they get decreased. And then some of the adverse impact of the humans, it includes liver dysfunction, cardiac diseases, and cancer, immune disruption, bone marrow suppression. Then cancer drugs, embryotoxic they are, and teratogenic effects were seen for fetuses. And antidepressants, they treat mental illnesses normally, but hypoglycemia, sexual dysfunction, and growth dysfunction is also correlated with the antidepressants. And surprisingly, for the antidepressant, increased rate of suicides are also correlated with the use of antidepressants. It was quite surprising for me when I first read it. But then anti-inflammatory analgesics, they are commonly we use it for as painkillers or antipyretics, but cardiac diseases and hypertension, mental health issues, they are related with it. So actually here, this is antimicrobial resistance. It's a hot, like a burning issue of today's world. It actually poses a threat to the global health setup, because there's a review written by O'Neill, an economist from the UK, I think, and he mentioned that by 2050 we will be having no effective antibiotic to treat a small scratch on our skin, and it can lead to death because we were unable to treat it. And here in my research I am focusing on antibiotic resistant bacteria to apply them to work for us. Like antibiotic resistant bacteria, they have the ability to degrade antibiotics, so they resist like the water which is coming from the pharmaceutical industry. For example, I am specifically focusing on ciprofloxacin antibiotic. So there are certain bacteria in those in that water which is actually surviving the highest concentration of ciprofloxacin, which is a very lethal antibiotic for them, but they are surviving, and it shows that they are resistant and they are degrading actually. So we developed two technologies, wetland technology, what we called. So antimicrobial resistance is actually a health security threat. These are my objectives, like treatment of pharmaceutical wastewater via lab scale constructed wetlands, and treatment of pharmaceutical wastewater via lab scale trickling filter. And then I have isolated some of the bacterial strains from both the systems, and then I compared the comparative analysis of both technologies. Now we will see, like this is my methodology. I took some samples of wastewater from the industrial effluents, and then here we isolated some bacteria from the untreated sample from the wetland and trickling filter using culture dependent methods. Like we cultured it on Newton agar media, and then we sent it for 16S ribosomal RNA, which is yet to be done because this all data is from preliminary research data. So it's in progress there in my lab. And then we running the samples on wetland and trickling filter. We collected the samples after running on those technologies, collection of samples and addition of new samples weekly. For 12 weeks I ran the reaction, and then we analyzed the sample on spectrophotometry. Like chemical oxygen demand was measured via spectroquant and prior and post treatment, and OD readings were taken on microplate. So here this is wetland technology. Like here you can see there are 4 cement parts filled with gravels, sand, and plants were planted there. These are the Typa latifolia plant, we can call it cattail in English, simple. And the microbes are actually in association with the roots of these plants. And from here in the first, here we add the sample, and with a slow flow rate it's coming down to the second one. In the first one there are only gravels and stones to trap the physical particle, like the large particles. It is like a physical removal, right? Sedimentation, filtration, and precipitation, these are the underlying processes in wetland technology. And then in the second one there are plants, there sand and soil is there to support the growth of plant, and gravels are also there, some sand is also added. So adsorption, why heavy metals like heavy metals and phosphorus, they adsorb onto the solid particles there. And phytoremediation, plants also take up some of the contaminants from the water, so phytoremediation is also there in the wetland. And then the bacterial decomposition and nitrogen transformation, already we know that bacteria transform the nitrogen which are in association with these plants. So this one is all about wetland technology, and it's constructed lab scale wetland. And here at the post stage there are no plants. The second and the third stage, they are same, they are only like the augmentation of the second stage. And the fourth one is only, it contains sand just to filter out more. And here it's a collecting tank, CT, it's a collecting tank. We run the sample here for 1 week. Like every cycle it completes in 2 days. Then we collect the sample here, again we run it, and like 4 times we run this sample and on the same, and then we analyze it. I think it's not working. Okay, This is trickling filter technology. Like you can see a vessel here. This is the outlet of the vessel. Like this is a suction pump there. It's like we put the sample here in this container, and then the suction, through the suction pump we add the sample from here. It's trickled down like a shower. It's like a shower. It's trickled down here, and here in the vessel inside there are stones which are covered with the biofilm of bacteria. Like how we did it, we actually grown the bacteria on media, and then we added the media containing bacterial strains on these stones, and for 1 month we ran the tap water, and then the biofilms developed, and then we added our sample. Like the underlying processes in trickling filter technology are physical removal, as you can see, and oxidation via aeration. Like the sample is trickling down and showering from the upside, so aeration is there and it's oxidizing actually the reduced compounds. And then bacterial remediation, like in this one. Now these are the results. Like it's significant reduction is there. For example, for trickling filter samples, first the ciprofloxacin concentration was 75 mg per L per liter, and then after 1 week it was reduced from 75 milligram to 7 milligram. Actually, it's quite significant, I think. And then in the second week it was 6, and then in the fourth week 6, 10, then in the fourth week 10, and then 9, then 11, then 16, and then come 16, 25, and then 27. Now you can see here it's increased there. It's because the trickling filter technology, it's mainly dependent on the outside environment. Like in the first 2 to 3, then the first 2 to 3 weeks, the environment, like the weather was hot, so the bacteria was quite active. But in these days, like we would add sample every week. So in these weeks the bacteria are quite dormant, because the temperature, yesterday I was checking in Pakistan, the temperature was 12, so it's quite cold there, and the bacteria is quite like we can say dormant, in dormant stage. And that's why these are increased value here. Now for the wetland samples, we can see from 75 in the first sample, untreated sample, it came to 2 on the second one. So it was quite hard in this week, and we can see here like so many reduction is there of ciprofloxacin, and then like from 86 in the second sample, it also came to 2.8 milligram per liter. Now this is the chemical oxygen demand reduction via wetland. In the first week it was 61. Like chemical oxygen demand, it's not only includes ciprofloxacin, it also includes nitrogen, sulfates, and other contaminants. So it's a whole value. Like so it's 61% it was reduced, and the most significant one here is 85.50. it's the highest in the wetland. For the third week it was quite hard that week. And then chemical oxygen demand reduction in wetland, it was 95.24% reduction in the 9th week here. So there are some like key takeaways or summary. Pharmaceutical contaminants pose health risk to all living forms, as like we mentioned about the antimicrobial resistance. And then but here I compared the two technologies, and I found that wetland technology is efficient, it's affordable, easy to handle, and eco-friendly, because it don't need any technical assistance, no need of electricity, no need of anything. It's quite eco-friendly. And then trickling filter, it actually includes the like we can say the suction pump, so it needs electricity and technical needs, and it's reliable and simple, but the technical needs actually overweigh its efficiency. So we have collaborated with the industry in the Pakistan, so we will be proposing the wetland technology to them, because they cannot operate trickling filter system there. And its cost-effective wastewater technologies, I think, must be installed at every outlet, like in industries, household, and healthcare, because it's very cheap. And then there are two Like the senior, these are my recommendations, like the senior and young scientists experienced in health security and leaders from the diplomacy must work in collaboration on developing strategies for the wastewater, because water is something very basic to our life, and we are wholly and solely dependent on water. Water is life for us. So, and youth must be the part of the working groups on local, national, international levels, because I thank you GYSS, because it's a wonderful platform here to collect together young scientists, and we interacted so well on the GYSS platform. And it was my recommendation in my country as well, that because future belongs to youth, so youth must be a part of every working group, like if it is in UN, it is in Singapore, everywhere in the world. So youth must be there. Like if you are taking decisions regarding future, so youth must be a part. Aknari means like my supervisor, she is a vice chancellor now, Dr. Safia Ahmed, and then my co-supervisor, Dr. Tahir. He actually got PhD from Washington University, and now he is working in Pakistan, and then some of my fellows in my lab. So thank you guys. It was all about it.
Thank you, Ms. Kasim. We invite Prof. Liu to moderate the Q&A.
Please. We thank Nassim for the wonderful talk. So now is a Q&A session. If you have any questions, please raise a hand. Or maybe just come to speaker, come to microphone directly. Yeah, please, yeah.
And thank you very much, and thanks for the presentations. And I'm home from Australia, from Australian National University. Also, I'm doing research on indigenous mathematics, but I used to work in the lab, and I'm also doing wastewater treatment. So I just have maybe two quick questions for you. So the first is that how you design your control group, or do you have a control group? Because it may be the biofilms, or it is maybe the filter function that of the silt, maybe not the wetland function to degrade the matter or the chemicals. So it is my first question. And the second question is that because and is these chemicals being extracted by the plants, or is these chemicals has been degraded, or do you have like any that further experiments like to test the concentration of the or on composition actually of the chemicals? Thank you.
Okay, I'll hand it to you. So the first question is, how do you do the control experiment?
The control experiments, actually we first, for the 3 months, we run the tap water on the wetland, and we analyze their values as well. Like tap water is clean water. Then it was actually our control group, like we compared then. Then we ran the industrial sample, and we compared both the results, and we found out that there are a lot of chemical oxygen demand. Like the previous sample I used, it has above 8,000 chemical oxygen demand. And in comparison to the tap water, it has, I think, 300. So we compared it. And the second question, you asked about the composition changes. So we are going for the soil analysis, from the plant tissue analysis, like how they are taken all the things, and where they aggregated in the plants. So we will be going for it, but it's in progress research. So for the composition.
Thank you for the question. Okay, this gentleman, please. Yeah.
Yeah, my name is Leo. Thank you very much for your talk. It was really interesting, and also a very interesting topic. I was interested in if you can scale up your wetland technology system, how do you see it to be most efficiently deployed in order to filter out pharmaceutical waste? Thank you.
Okay, so actually it was a project of the Washington University. We installed a large-scale wetland as well in our university, because there were some issues of wastewater. They were coming from the university, and you know that labs of biology and chemistry, it contains a lot of waste chemicals. So the government actually directed us to clean your water. Then we sat there and we thought that we have a plan, we have a technology of wetland. Then we installed a large-scale wetland in our university at Quaid-e-Azam University, Pakistan. We have a wetland here, and now it was actually for the purpose of treating household water. And now we are changing that technology, and we are using that technology. We want to check if it is efficient to treat the pharmaceutical water or not. So we will be changing it a bit. Like it's now working very well on the household water.
Cool. Thank you very much.
Yes, please.
Hi, my name is Chiral. I'm from the University of Cape Town in South Africa. First of all, thank you for your presentation. It was really lovely and well done on the COD removal. It's quite impressive values that you've reported. I have a few questions that I would like you to clarify. First question will be on your multi-species biofilm development. Can you please explain more on that, on how you selected the microorganisms or how you selected your community based on the waste or based on the components that you're trying to remove from the waste? Your multi-species development.
Okay, multi-species biofilm, yeah. Multi-species biofilm, like we ran, as I told you, that we developed the culture first. So how we developed the culture, it was not selective. It was not selective because you cannot go for a selective biofilm in environmental microbiology, because if you are going to the environment, all the things will be mixed. There will be a blend of bacteria. You will be addressing it. So we isolated some of the bacteria, but it was a mixed culture and the nutrient broth, and then we put it on the stones in the trickling filter, and for a month we ran water and then it developed biofilm. Now I have isolated like 5 strains of bacteria from those biofilms, and now we have sent it, but mostly on biochemical characteristic there seems that there are bacillus species there, and because bacillus are inherently resistant to antibiotics, so bacillus species are there and staphylococcal species is there.
My question is, where did you sample?
From the industrial effluents.
From the industrial effluent.
Yeah.
Thank you. My second question will be on your loading rates, your COD loading rate. Does your biofilm have a specific tolerance, or did you do any initial loading rate studies on your COD tolerance?
No, like when we took the sample from the industry, we directly measured the COD of that sample because we didn't know how much it was. Like then we measured it and it was about 8,000, and then like it was not a synthetic sample, it was an organic sample from the industry outlet, and then we measured it, so we came to know that it was about 8,000.
Is the COD value of the COD of your industrial waste the same at each sampling time, and do you load the same COD at every start of the treatment?
Yes.
So your microorganisms are adapted at that starting loading rate?
Yes, we can say that.
Okay, that's impressive. Second, whatever number question. My other question is, you mentioned that you add a fresh culture every week, or is it the fresh
No, it was not a fresh culture, but a fresh sample. Like we took a lot of sample, like 30 to 40 liters of sample from the industry, and then we were changing the sample every week.
Changing what?
Changing the sample, like we ran the sample for 4 cycles per week, and then we collected the sample after treatment of 1 week, and then we added another sample from the same tank, but another sample. Just like in wetland technology, you are going to run the sample for 12 weeks because during the treatment, the biofilms are going to develop.
Okay, all right.
So you will be like measuring step by step for 12 weeks, but 12 weeks, I think it's ripened there. Then we can have a fruit.
Last question. So you're operating at ambient temperatures. You don't control your temperature at all.
Yes, because it's outside from the lab.
Yes, your rates are quite impressive
Yeah, yeah.
for ambient temperature, but then you notice that at seasonal changes, when it was
Yes.
getting cold, your microbial activity was quite low. And for industrial applications, do you think that's something industry will have to consider and implement a heating system? And
Exactly.
what will that do to the cost? Because you're very confident with reducing cost, but applying a heating system might shoot up your cost as well. So is that something industry will easily accept, and also considering the area of your industry, because it could be at some very cold areas?
Yeah, I got your question. It's a very interesting question. When I first took the course and the research then, so I thought that it might be the problem for me. But then we discussed it and we proposed a solution. Like every industry, if it's working, a lot of heat is produced in the industry. So we can, like for the bioremediation, it was once used in Pakistan, that we proposed like a duct system. Like the heat from the industry itself, it will be directed in pipes through the wetland. So it will be heating the water. So I think it's also cheap.
All right.
From the same source, heat from the same source, we will be using it for same industry.
Thank you so much.
Thank you for
Welcome.
the question. Maybe you can bring the technology to Singapore. Here is always hot. Okay, so now let's take the last question due to time constraint. This gentleman, please.
Yeah, so at the end, aren't you growing antibiotic-resistant bacteria?
Like,
I mean, you said that you have bacteria that degrade the antibiotics.
yes.
So I wonder if you are not growing antibiotic resistance in your wetland.
No, actually there are two projects. One is running by my fellow. So she is actually looking for the efficiency of wetland, if either the wetland is efficient to remove the antibiotic resistant genes like ARGs. There's a global pool of antibiotic resistant genes. So she is looking to it, like how much wetland is efficient to remove the antibiotic resistant bacteria in the genes. And I'm looking for the drugs removal. So we are like we are taking it side by side. Yeah.
Thank you. So in parallel with this on-site meeting, right, we actually, the video is also projected online. So allow me to take one question from the online pool, just to say hello to our online participant, right? So the question is, are you planning to compare the microbial composition of pharmaceutical companies from different locations, maybe at different time as well?
Actually, we got sample from one industry initially, but now we want to like broaden the horizons of our samples. We will be looking for another sample too, and it's very good to consider it. I'll be considering it.
Very nice. Let's look forward. This wetland technology will change the world. Okay.
Yeah.
Yeah. With that, I want to thank the speaker again for the wonderful talk and for answer all our questions.
Thank you.
Thank you, Prof. Liu and Awais Kasim. Now I shall invite Prof. Liu to present the certificate of appreciation to our presenter. Thank you, Prof. Liu and Awais Kasim once again. This concludes the series of Young Scientists presentations. We will now be setting up for the next segment. And good morning once again to those who might have joined us from the small group sessions. I'll do a quick recap on the housekeeping announcements that I have also shared earlier today. So first, we'd like to hear your feedback on your experiences over these few days. So we have actually sent you a feedback form via email yesterday. So do assist us to fill up the feedback form. And also, you can find us on social media. And so follow us on social media if you have yet to do so. So you can find us on Facebook, Twitter, and also Instagram. Up next, we have the panel discussion on science communication. The panel features Sir David Klenerman, Prof. Alison Woollard, and Prof. Valerio Scarani. Moderating the session today will be Mr. Jin Tan, Assistant Chief Executive at Singapore's National Library Board. So let's warmly welcome the panel and our moderator. And I hand the session over to Mr. Tan, please.
Hi, good morning. We're not ready yet because I need some technology training. Okay, so we'll start at 9. Is that all right with you guys? Yeah. How are you guys feeling today? Yeah? Some of you look quite... Can you? Hi, how's everyone? You know, I heard that we've lost half the audience after 4 days, but you guys are here because you're committed. So this is what I need you to do before we begin the session. We're going to have a Q&A session fairly shortly, and I really don't like to fake questions. You know, moderators, they come up with a question, and I think that's quite tragic. So could you guys start to create questions? Yeah, you know the Slido thing, right? Do you guys know the Slido thing? Yeah. So if you have any questions on communication that you'd like to ask, start formulating them right now. It's good to formulate them early so that they'll sound fairly intelligent. Yeah, so can you guys do that? Yeah, okay, thank you. Okay, let me do a quick introduction. My name is Jean Tan, and unfortunately that title is gone. The one that you heard earlier is gone. Okay, so I'm now the Chief Librarian, which is a much nicer title, and the Chief Innovation Officer at the National Library Board. Yes, only one person applauded, yeah. Thank you, sir. Yeah. You're very important here, right? I heard, yeah. Okay, so thank you to the local god for applauding me. Let me introduce the topic for today, which is science communication. I think communication is, I've heard it's important, not just the science community, but also the stakeholders, including children, because they could be the future scientists or consumers of science. And also with comms platforms now becoming so complicated, and even the consumption patterns becoming so varied, I'm really interested to know how our esteemed panelists think about that. Let me do a quick introduction of everybody, and then we'll go through the format. So first, Sir David Klenerman, the Royal Society Professor of Molecular Department of Chemistry, University of Medicine, University of Cambridge, sorry. I'm sure you would run a University of Medicine if there was one. Yeah. He received the 2018 Royal Medal by the Royal Society and shared the 2020 Millennium Technology Prize and the 2022 Breakthrough Prize of Life Sciences with Sir Shankar, I believe. And then let's give him a round of applause, shall we? Yes, and then we have Prof Valerio Scarani, who is the Principal Investigator and Professor at the Center for Quantum Technologies, National University of Singapore. He has done significant efforts, I think, in engagement, and I believe there's a publication called 6 Quantum Pieces with NUS high school students, yeah. Did it sell well?
Pretty well when we promoted it.
Well done.
It's going little by little. It was not, of course, a bestseller on the first week.
But it's going well.
It's keep going.
Nice,
yeah.
nice. So are you rich now? Are you? No.
No. Actually,
not from that.
I gave the royalties to the students. I didn't need them.
Oh, you're so wonderful. Yeah, let's give him a round of applause just for that. Yeah. It's very nice to have generous and successful scientists. And over here, ladies and gentlemen, we have a television celebrity. If you have stalked her like I have before the session, you have seen several videos of her online, and she didn't remember this, but I remember she had a stunt double in one of them who did like backflips and somersaults. She's not going to do that today, ladies and gentlemen. This is Prof. Alison. Are you going to do that? No. Okay, yeah. So we have Prof. Alison Woollard, who is a fellow in biochemistry, Hertford College, University of Oxford. She's also the associate professor in the biochem department, University of Oxford. She was awarded the 2015 JBS Haldane Award and appointed a University of Oxford academic champion for public engagement and research. Thank you, Prof. Woollard. Let's give her a round of applause. Yeah. Okay, so now this is how it's going to work. I'm going to ask each of the panelists to talk about their experience in science communication. They'll take about 10 minutes or so. And while they're doing that, do send in your questions as they come to you. I will also be asking all those present to go to the microphones. You can see them on the left and the right of the hall to pose your questions. Yeah, priority will be given to those who ask the questions in person, because I think courage should be rewarded, don't you think? Yeah, I suddenly sense a dip in courage when I said that. Yeah? Okay, so live your courage, yeah? Sir, you should ask a question. Yeah, you look like the questioning kind, sir. Yeah, you have a beautiful open face. Yes, you. There's no sir behind you. Yeah, okay, that's a Sir Dave here. But so can you ask a question? Can you like start formulating one? Yeah, let me see you working. Yeah, no, not now. Yeah, not now. Yeah. Formulate your question. Okay. So, ladies and gentlemen, he's been assigned the first questioner. Let's give him a round of applause. Yeah. Should never sit in the center of the room. Yeah. Okay, so we're going to get our folks to start sharing their views on this. We're going to start with, did I say Sir Dave with you first, right? Sir Dave, yeah.
Alison was going to start.
I thought Alison, you're going to start, right? Okay, see, I just got the order wrong. Yeah. Alison, would you like to share with us your experience?
Yeah, of course. And I'd like to thank you, actually, Jean, for such an engaging introduction. And already the audience feel that they're part of this conversation because you've picked on some poor soul already. And everyone's thinking, thank God that wasn't me. But they're also thinking, oh, actually, maybe we've all got permission here to ask whatever questions we like. And so I think that's a really important ethos and atmosphere. So thank you for that. So what I want to say here, so what I don't want to do is completely steal my thunder because I'm giving a talk later on about science communication. And I don't want you all to think, okay, so we know what she's going to say, so we're not going to come. So I thought I would just make a couple of comments about why I think that communicating our research is particularly important at the moment. And then I would tell you a little bit about my own kind of personal journey in that space. And so the reason I think science communication is important at the moment is because we need lots of scientists. There are lots of big challenges out there. There's some great successes, but there's a lot of work to be done. There's huge challenges to face, and science offers some really incredible solutions to some of those problems. Not just science, but science is a really important part of that. And so we need to encourage young people from all over the world to go into science. And science only works if the people doing science are open-minded and collaborative and diverse. And I think that the more engagement and communication that can happen across many, many different platforms, there's more chance that science will be truly representative and involve endeavours from many, many different kinds of people, different communities, and so on. So I think that's one really important reason to do engagement and communication. And the second reason is I think it's really good, excuse me, it's really important for trust. I think if you have a really in-depth conversation with someone about how your science works, how you do the experiment, what it means to make a discovery, to build evidence, to gather weight of evidence around a particular idea. And if you talk about the fact that sometimes science is iterative, it's not that you get the answer and that's the ultimate truth forever. It's sometimes that you're limited by the technology and then you have a theory and then 5 years later you have better ways of doing the experiment and you find a slightly different answer, and that's okay. That doesn't mean that the first answer was wrong or that there's some dishonesty involved. It just means that's the way science is, an iterative process. And I think it's really important that people understand that, because that helps in trust, because scientific advice changes as we find out more and more. And trust in science, I think, is very, very easily eroded. And you only have to look at all the conspiracy theories that you can find online to know how easily trust in science can be eroded. And, a good example would be the anti-vaccine movement. It's a very, very powerful voice. And it's a difficult one they're a really difficult community to engage with, actually. But I really strongly believe that you have to try. And just shouting the facts is not the way to do it. Because people are skeptical and have views against a particular bit of science. Sometimes not because they're stupid or uneducated, but because maybe they don't have the tools in their culture to appreciate and to accept the science that we all accept. And so trying to find out why someone has the views that they have is really important part of that process, rather than just going in there and saying you're wrong and you need to change your mind. So that's sort of my view on why science communication and engagement is important. My own journey in it is, I think the first time I ever did any science communication was when I was a graduate student and I was working in a cancer research institute in London. And all graduate students had to go and give a talk at there were a lot of volunteers that raised money for cancer research, and they typically have shops where you sell second-hand clothes and what have you. And so the volunteers run these shops, and these shops are really important for raising money for cancer. It's called Cancer Research UK. And so the volunteers can sign up for a talk. And so I was sort of allocated my talk. I'd never done any kind of science communication before. And before the talk, there was a training session. And the training session was run by a media company who came in. It was really, really good. And they filmed you doing this talk. And then they looked at the film with you and sort of said, this is actually really terrible. Really, really terrible. Because of and then they took you through it almost kind of frame by frame, which was really felt really scary at the time. But they did it in such a kind of funny and nice way that it felt very inclusive. And so I sort of improved, I guess. And then I gave my talk. And the talk quickly broke down into a conversation. Because there were about 20 people in the audience, and there was that, so you could talk, you could have quite a good conversation. And I realized that, the people that had come had a hunger to understand how basic research in cell biology can ultimately influence cancer therapeutics. And they wanted me to help them join the dots with those things. And it was I loved it. It was a great conversation. And then after that, I sort of volunteered for more and more things in that kind of area. And then when I started my own lab, eventually, I got some funding from a particular funder that wanted to do a film of me talking about my research and why it's important to have got this particular funding and blah, blah, blah. And I did that, and they put it on YouTube, which was quite new at the time. And it was that video on YouTube that the Royal Institution picked up when they were looking for somebody to deliver the Christmas Lectures, which is a BBC TV show back in 2013. So it goes to show that you do these little things, an opportunity comes up and you take it, and you never know where that's going to lead to. It can, just for very serendipitous reasons, someone can pick up on something you've done and you build on that. So I'm going to end, but that's how my journey in public engagement started. And I'm going to end by saying that my advice is don't turn these sorts of opportunities down. Have a go, do them, because you never know where it's going to lead. I think that's me done.
I think it's wonderful. I especially like what you said about how your talk became a conversation. And I think that's really good. We should try and engineer one later. What do you think? Yeah, let's engineer a conversation with one terrified member of the audience. Yeah, I'm looking around. I'm looking at someone with the best hair. Yeah, shall we engineer a conversation later? Madam, shall we do that? Yes, I'm looking at you. No one's better hair than you. Yeah, so we'll do a conversation later, shall we? Yeah, okay, so we have one designated questioner. We have one designated conversation starter. Okay, so we'll do all that, and then we'll go to Slido. Okay? Yeah, so now let's go to Valerio. Tell us about experience in science communication.
Yes, so it's pretty much serendipity what happened in my case. So I was at the start of my PhD in EPFL Lausanne. And because of some great teacher that I had for quantum mechanics, PhD in physics, by the way, at some point I developed a kind of understanding of how would be the good way to explain quantum mechanics. And I thought, okay, well, is it really original or not? I don't know. But what I try to do is to advertise a talk, improvise and advertise a talk to everybody who wants to come, maybe my fellow colleagues. And so at the time, speaking of 1996, this was the time where some of my technologically inclined colleagues were telling me that, you can start sending messages through a computer, okay? And I was surprised. Why would you use a computer to send messages, right? So that's the time. So of course I printed these, colorful things, a piece of paper, put it on the notice board of the university saying on that day, that time, Valerio Scarani, completely unknown by the way, would explain quantum mechanics. Okay, and some people turned out. It went okay. I was of course scared that some of my professors would turn out and then trash me. It didn't happen. Actually, I think one of the professors did show up and liked it. And I kept doing little by little these kind of things with friends outside in different communities and so on, and people seem to like it. Again, the next breakthrough was, again, a serendipity event where some younger friend of mine who didn't have a car asked me if I could drive him and two of his friends to the book Salon du Livre, the book fair in Geneva. So we drove there, and then I was thinking of going around this book fair with these young people, JC, as we call them, so the high schoolers, and talk with them. It turned out that the whole goal of the operation was that their girlfriends were there. So as soon as I arrived, they told me, you know what, let's meet here at 5, and they disappeared. So I find myself there in this book fair thinking, well, okay, well, fair enough, I go around and see what happens. And I found a scientific publisher. I started talking to them and say, yeah, I have this idea on explaining quantum mechanics. Would you be interested in writing something and so on? It turned out that these people had a famous scientist, a famous French scientist was the editor of their scientific collection. So they told me, yeah, write your proposal to this person, Jean-Marc Lévy-Leblond. So I wrote it by email already. I could use that tool by now. And this professor that is famous in France, also very much engaged in communication of science, was very happy about my approach. That led to my first book on quantum that in the French world was quite successful, and later was translated into English. And by the time it was translated into English, I asked someone that I knew by then to write the preface, someone called Alain Aspect. The name sounds familiar, maybe, because he got the Nobel Prize this year. He was the first person to do what is called a Bell test, a test of Bell inequalities. So now I have even a book prefaced by a Nobel Prize winner. This was many years ago, of course. And then, okay, the story goes on. I come to Singapore, and before coming to Singapore, also I did in a school some things called, well, again, I don't know exactly the English translation. Projet de maturité would be in French, which is the graduation project for JCs. Okay, so something like they had to do a kind of small project. At the time, no ChatGPT, of course, so they had to do something. So I invented some exercises of quantum mechanics, simple, this time with mathematics, simple exercises for them. When I came to Singapore, it was also the time where this NUS high school had started, is a kind of elite school for people devoted especially to science and maths. And some of these young students wanted projects, again, wanted to do something and so on. I was trying to convince them that, I mean, for me, giving a project in research, I'm doing theoretical quantum mechanics, it's not that easy. But I told them, well, what we can do as a project is you take these exercises that I've written, you solve them again, you do it better, you write them as a text, and together we compile a book. And that's the origin of the 6 quantum pieces that was mentioned. So that's the second, the fourth. After that, well, I went more into research, so I did only occasional things. It's not, until recently, right now, I start teaching a new class in NUS called The World of Quantum, where I'm trying to combine the best of my two books, because in NUS now we have kind of migrated to a kind of more American style, where the sciences and arts have a common curriculum at the start before separating. And in that context, therefore, now I'm teaching quantum mechanics without mathematics to a very interesting audience that goes from data scientists to, there's even a student from Chinese studies in there. There are some psychologists, some physicists, and on. That's a new attempt. Let's see how it goes. I don't have any feedback yet. Yeah, that's pretty much my story. And I mean, the message I've been always trying to push forward is that there is a way to present quantum mechanics in a way that the fascination and the difficulties and the conceptual interest strikes you directly, without going through writing down complicated mathematics and Schrödinger equations and waves, or even things like black body radiation, that to understand it you need a bit of physics. right? There are ways much more striking nowadays because of experiments that have been done, including this one of the Bell test by Aspe and others, that really put you in front of the quantum reality and say, wow, right? That's how nature behaves. Okay, that's my parabola.
Thank you so much. I like what you said about how you have to teach students ranging from data scientists to those who are involved in Chinese studies. There's a very good question that you have to answer later on Slido about how science should be communicated with less technical terms. Yeah, so we'll come to you for that later. So now we have Sir David. Sir David, tell us about your experience in science comms.
I think compared to my other panelists, I'm a novice at science communication. So I've done, I think, 3 types of science communication. So the first type is if you're lucky enough to publish a paper in a high-profile journal, there might be a press release, or when we won the Millennium Technology Prize. And in those examples, the requirement is to really explain what you've done in very simple language so the newspapers can write it up. And that's actually quite a challenge. You're used to thinking about the details of the methodology, and to have ways of explaining the DNA sequencing that are very understandable was quite a challenge. But that's the first example. We also had some live interviews, and again, we really needed to have very clearly in our head how we were going to explain the technology. And it's challenging because we use words like DNA, nanometers that we're used to using all the time, and you suddenly realize people don't know what DNA is, they don't know what a nanometer is. So we really needed to make sure we got the language right. And the reason I think it's important is we're really advertising what science can do. We're trying to convince the public that they should spend some of the government money should go to science, and it shouldn't be go for better roads or increasing other people's pay. So it's important to get these arguments right and make a strong case for why people should be funding science. And so it's very important, but it's very challenging. Well, I find it very challenging because you're using a almost different language to explain what you've done. The second example is quite similar. We're going to schools and try and present our science. And again, we're trying to advertise, I'm a chemist and this is what a chemist can do, but we're also trying to demystify the process of being a scientist and explain to people exactly what it's like if you do a degree in chemistry to do scientific research. A lot of people really don't appreciate what it is to do research. They're used to doing practicals, they follow the recipe, and then if they do the experiment well, they get the right answer and they move on. And as you all know very well, it's entirely the opposite. You don't know how to do the experiment, and if you get to do the experiment, it doesn't work, and you have to figure out why it doesn't work and go around that loop lots and lots of times. And again, there's similar challenges, maybe less challenges around the language, because you're talking to people who have got a scientific background. And my third example is probably the most challenging. So we do a lot of research on neurodegenerative disease, what's the cause of Alzheimer's and Parkinson's disease. And I've been to a conference and I've been put in front of people with Parkinson's disease. And the first thing is you go to these meetings and you've studied Parkinson's disease, but you... So this conference, that there was a parallel session of people with Parkinson's disease and scientists working on basic research on Parkinson's disease. And then we had some joint sessions. So the person talking with Parkinson's disease, they had to cut back onto the stage. They couldn't work forwards, but Parkinson's disease, you have problems with your movement. So they were back onto the stage. One of the speakers' arms was waving around wildly because they just couldn't control their limbs. And it's very personal. You're suddenly, right in front of you is the person with the disease that you're working on. And then it's quite a challenge to know how you present your research. You want to present it, you don't want to give people false hope, but you don't want to be too negative. And so it's quite a challenge in front of someone with the disease, how you explain your research and what's the potential for your research to solve the problem that they're living with. And I'm not sure I had a very good way of doing this. Certainly, my approach was to take the DNA sequencing and saying, this has been very powerful, and we're trying to repurpose the DNA sequencing to solve your problems. So that's my limited experience in science communication, and I'm hoping I'm going to learn from my other panelists how to do it better.
That was actually one of my questions that I had prepared in case I couldn't find anyone to ask questions, which was what was the toughest audience you had to crack? And that seemed like the one. So if you don't mind, I'd like to pursue that a little bit. How did that session go, and how did you think it went?
I think the people are very, very engaged. You mean that you sit next to them, they've read all the literature on Parkinson's disease. And so certainly our pitch was, we're taking a high-tech approach for the first time to try and solve your problems. And this is how far we've got, and this is what we're going to do next. And I think it went well. We weren't promising a cure.
You're responsible.
So I think it went well. I think it's more difficult when you think you're starting to move towards a cure, how you communicate that. But I think, you know, we're very early stage, so we just say we're starting. Sure. It's a different approach, and I think it was well received.
So David, mind me asking, so what was the objective of that communication session? I'm always curious about that.
So the one example is we have a clinic in Cambridge where people have Parkinson's disease, and every 6 months these patients come in, and the scientists involved in working on Parkinson's disease tell them about the research they've been doing on Parkinson's disease. But these patients donate blood, sometimes they donate cerebrospinal fluid, and it's a way of feeding back how those samples are being used. And that's the purpose.
Absolutely.
And so it's just to keep them engaged and encourage them to keep on coming back and allowing people to take blood and take cerebrospinal fluid.
And that's what I think, Alison, you're talking about, trust. I mean, that is the ultimate level of trust that you need to get from someone you're communicating to, for them to give you blood. Yeah, that's the ultimate level of trust, I just want to ask the panelists before I go to, are you ready? Oh, he's ready. So why don't we let him go first, Then I'll come back to you. Okay, sir, can you introduce yourself and, maybe your illustrious career?
Sure. So I'm Patrick from UCL, and my research is in medical imaging, MRI for Alzheimer's and neurodegenerative diseases. And my question to the panel is related to presenting to a broad audience, or high schoolers, but it could be even the general public. Because oftentimes I find that when you try to explain complicated concepts, like say quantum mechanics, it's much easier to dumb it down rather than respecting the intelligence of your audience and keeping the technical intricacies in simplified so that people can understand it or follow it. And oftentimes it's a language thing as much as it's a concept thing, where using the right words makes it possible, but finding those words is very difficult. So I wonder if you have some insights on perhaps helping us find the right words to share our research with a broad audience without dumbing it down.
Thank you, Patrick, And if I may add, there's a question on Slido that's quite close to that. So I'm going to read that out from Mr. Rahu from United Kingdom. It is often suggested that science should be communicated with less technical terms. Could you suggest how that can be done effectively for diverse audiences? So I'm very interested in how, Alison, you'd like to share your views on that. Then Valerio, I'm interested in how you've been doing your lectures for that whole range of students.
Yeah, so first of all, I think I acknowledge the challenge involved in it. But I think it's really worth spending time on, because actually I hate the idea of dumbing down, because then you lose the beautiful idea if you do that. And there's no reason why the average person can't follow your beautiful idea. It's not that we're kind of, super mega intelligent geniuses. We're all kind of normal people at heart. And so it's not a case of dumbing down the concept. It is about finding the right language. Sometimes it's useful to use some kind of analogy, some kind of story that kind of portrays that concept, and then swapping the characters a little bit, saying, well, actually, this is this. But you do have to be quite careful with that culturally. Sometimes you have a little metaphoric story that somebody from another culture, they've got no idea what you're talking about at all. So you do have to be quite careful about that. But for me, when I put together a talk for a different kind of audience, the first thing is to really understand who the audience are. And that helps a lot. And then you have to, in your head, I think you have to try and get into that journey with that person or those people in your audience. So you're aware as you're speaking that you're taking them on a journey. So be in that journey with them. And it can be quite difficult to do that when you're nervous and you've got your thing to say and you want to say it. But you have to try and, I think, slow down and stop and think about how it is, what the words that are coming out of your mouth, what they mean to those people. And then if you think people are not following you, then go back and, try and have another go, try and change it. So yeah, never be afraid of the conversation, I guess. Yeah.
Excellent. Prof?
Yes. Well, I can only speak of my own experience, right? So unfortunately, I cannot help for that. But so when it comes to quantum physics, fortunately, there are experiments, right? So, and the reason, if you want the point of view that I was trying to adopt, we can discuss infinitely long later, is what led physicists to formulate such crazy theory? I mean, at some point, of course, at the beginning, there's a whole story, it's very complicated and so on, but at some point, in order to believe these things, right, with whatever, uncertainty and entanglement and whatever, this must be based on evidence, right? It must be something you can see. And then what I present to the people is what is the evidence that forces us out of our classical worldview. I cannot honestly introduce how to solve that problem. I mean, how to quantum theory is phrased. I mean, I'm trying to, but I will not go in with the students and others to the point where saying, so they have to trust me that now we have a theory that treats things differently than intuition, and through that theory we can predict stuff, right? And everything fits. That part, yes, I definitely cannot convey, right? Also, another very important thing when it comes to physics, at least I don't know in other fields, is that there are many words that come from theory, and they don't have, for instance, the famous word entanglement, right? Entanglement is a mathematical notion. Reality is not entangled. Reality is reality. And we describe that particular phenomenon with this mathematics called entanglement. The closest we have is called correlation. Now, that's another thing that I found, however, is that you said, and everybody says that we should use everyday concepts. But people don't know what is a correlation. I mean, if you don't know what is a correlation, there's no chance I can explain to you what's quantum mechanics. So there is, at least at this level, that's why, for instance, I refuse to explain quantum mechanics to primary schools. At some point I was asked and I said, let's start when people are 15. Okay? Because at least the notion of correlation, the notion of randomness, where does it come from, and so on, these should be notions that one is a little bit familiar with, right? Starting from TikTok, I can't explain quantum mechanics.
Interesting. I was going to ask you about TikTok. Let's get to that later. So, David?
So certainly in my field, it's quite visual, so we can use visual aids, which I think you don't need to use language. You can just show people pictures, and that's quite powerful if you can get the right picture. And then my other thing, I would say, certainly for us, it's not drumming down. It's much more distilling it down to the essence. And I think, you've invested lots of time in getting a certain step to work, and you want to put that on the slide, and you have to just try and distill it down to what's the essence, what I'm actually trying to tell the people. And certainly with public engagement, it's not the details, it's the high-level picture. So I wouldn't use drumming down. I think it's really important to really try and distill it down to its essence. And it's difficult, but I think it's a useful skill, not just for science communication, for writing grants. I know the Wellcome Trust, summarize your question in 150 words. You really have to distill what you're trying to do down to the essence. And I think we spend so much time worrying about the details that sometimes it's nice to come up and take a view from the sky and see what you're doing.
Yeah, so it's the top line essence. Okay, we have a lot of questions coming up, so I'm going to give you guys numbers because I'm getting confused myself, okay? So remember your number, yeah, and don't jump queue. Okay, so I'm gonna give you guys numbers, and then later we still want a conversation at some stage, yeah? With that lady and the ladies around the lady, yeah? In case the lady is not interested in conversation. Yeah, so the ladies around the lady should also be prepared, right? Are you all right? Yeah, getting prepared? Yeah. Yeah, so prepare to have a conversation with Prof. Alison later. Okay, numbers. Okay, so ma'am, you're 1. Yeah, the blue ma'am is 1. Okay, and then this lady here, you're 2. You're 2, yes, you. No, not you, the one behind you. Yeah, you're 2, right? And then the lady behind the blue ma'am is 3. This gentleman is 4. And the blue guy is 5. Okay, I'm so sorry. I'm coloring you guys. It's the easiest. Okay, so that's 5. Okay, then after that, we have 6, 7. Yeah, lady, you're 7. Yeah, ma'am, you're 7. No, I think you came later. 7, 8. I think you came after that. 9, and 10. Okay, sir, you're 10. Okay, so I'm just gonna call your number. I'm not going to go blue guy, blue ma'am again, okay? I'm just going to call your number and you just speak, okay? So anybody who comes after 10, we'll see what happens, okay? So can we go with number 1, please? Yes. Ma'am, number 1.
Hello, everyone. Good morning. My name is Mariana, and I represent Spain and Colombia and the University of Cambridge. I'm doing a PhD in stem cell biology and hematology. But I'm also an educational and science communicator on social media. And that's why I'm very engaged in this conversation. So I have 2 questions, but based on the situation, I'm gonna go for 1. Could you comment on the effect or relevance of the representation of minority groups or less privileged communities in science communication?
So minority groups, and there's also a question that I'm going to relate to that here online. And the question is, can you elaborate on the importance of diversity for science communication and how it influences scientific creativity and innovation? Yeah, so minority groups, diversity. I'm sorry I'm conflating it, but if you could arrange, I could address a range of topics for that, yeah. So would anybody like to address that?
I can start. I think it's really, really important because there's a lot of inequality in science. You know, you look around our best universities, you find a lot of white middle-aged men. You don't see so much, present company accepted, of course. You don't see too much diversity, and that's a problem for science because different groups have different ways of doing science. They have very different styles. They may work on different things. And so you need a diversity. You need to involve all of humanity in the effort in order for science to, you know, to work as well as it possibly can. So I think it's really important. So it makes sense then that engagement with science needs to involve a huge diversity because that's the way that you can inspire and facilitate people from a huge range of different kinds of societies to want to be part of it. And so open-minded collaboration is just absolutely crucial in science, I believe.
Are there any structural obstacles to that, do you think?
Yeah, there are, because, you know, and that's why it's important you were talking about, you're involved in social media. And that's so brilliant because it reaches a whole different group of people. And in many ways, it's more democratic. You know, I give a lot of lectures in London. Now, the people that come to those are not necessarily socioeconomically deprived. They tend to be middle class, white. I think that's a big problem. And increasingly, I see the importance of social media platforms in reaching hard to reach people. But you know, there are other things that can be done as well. I'm very involved in work that visits schools that are in economically deprived areas. I won't actually go and give a talk anymore at a British public school. I say no, because I think my efforts are best used elsewhere. So we can make those kinds of decisions as communicators, and I think they can have a really big effect on the outcome.
Thanks, Alison.
I mean, I would just comment that I've
David.
talked at public schools, and the public schools have gotten lots of local comprehensive schools, and so it has been much more democratized. So I've given a couple of talks, and it's been very impressive, you know, the range of people who've come, and so it almost provides an opportunity for them. You know, whole class came. So I think the public schools, to defend them, are actually trying quite hard to when they bring people in, to maximize the impact of that and bring in lots of local communities.
Yeah, thanks very much. Okay, I'm going to go to number 2.
Thank you.
Thank you so much, number 1.
Hello. Yeah, my name is Svana. I'm doing a postdoc at the Biophysica Institute in Spain. And my question is quite basic. So how much time do you think we should dedicate in doing science communication, and how much time should we actually dedicate in doing actual science? Because I have the feeling that, yeah, I'm spending more and more time to actually explain my research to a broad audience or potential funders, but less and less time actually to do my research, which is actually why I became a scientist. So yeah, what do you think on that?
How much time do you spend on communication? Yeah.
I mean, I would do 4-6 talks a year. I'm right at the low end, I would think, of the panel.
Yeah, and are you on social media? I've seen you on YouTube.
I mean, I'm not very good. My group put me on some of these things, but I would say I'm a dinosaur when it comes to social media.
Yes, but you're a dinosaur with a very good voice-over voice. I have to tell you that, Sir David. Yeah, you've got a very nurturing voice, and I can imagine you just voice-overing, you know, documentaries on science, yeah. Yeah, don't you guys sing? Yeah, he's got, sorry, I'm so sorry. He's turning red. Are you generally red? No. Okay. Moving on. Yes, Valerio?
I mean, I don't think I have a personal answer, but I have a kind of corporate answer. So in my institute, Center for Quantum Technologies, at the beginning, I was in charge of the outreach. And of course, I mean, you get overwhelmed, right? Because there's a website to update, what to put there, and so on. At some point, we hired someone. Okay, that's what we could do as an institute, right? So for my own research, well, I have sometimes to sell to grantors, and failing at it, by the way, if anyone is here from MOE. But for our research institute, it's quite big and visible. We have a person, Jenny, that takes care of, she has a physics background. She has been working on communication of science for many years. And yeah, she does a fantastic job. And from time to time, she comes to talk to us when we publish a paper, maybe in some important journal. She comes to talk to us, she interviews us, or she asks us for a write-up, and then we promote this thing. So that's the solution we found, because it is indeed a problem, right? For a kind of individual or for a small group that cannot afford to hire a professional person, it is a problem. And I experienced that at the start. Fortunately, in my context now, I have that. So unfortunately, I don't have that problem anymore.
You have institutional help, yeah. Very good, very good. Yes.
Yeah,
Prof, you don't need any help, do you?
I do recognise the problem. So, you know, I'm a researcher, primarily. Universities find lots of ways to distract their academic staff, though. It's not, you know, I think I spend more time in boring committees than I do doing public engagement, so... So there's that, but I do think it's an issue. I think if you can develop some activity that you can reuse, recycle wisely, that helps a lot. Working in a team where you're not just doing everything yourself, and you know, trying to get some help from your institution along the way. So you obviously, if you want a career in science, you have to do the science. But some people shift careers into public engagement at some stage. So if you might want to hedge your bets, but you've got to find time to do the science as well. And as we all know, that's a kind of relentless, never finished task. Yeah.
Yeah, and there's a question on media consumption. I'm going to assign number 11. Ma'am, you're number 11. You're 11, right? And I'm going to just ask whether you guys want to sit if it's not your turn yet. You have your numbers, right? Yeah? I'm sorry, this is like taking restaurant reservations, but if you like, you can take a seat if you like. And and rise when your number is called, and assume that you guys are science people, so there won't be any cheaters who will try and pretend to be a number that they're not. Okay, so the rest of you can take a seat. Yes, take a seat if you like. And sorry, what number was I at? So now it's 3? Now it's 3, okay. So we're gonna do a 3, and then we're gonna do a 4, and then after that we're gonna have a little conversation. Is that all right? No, you don't want to converse, yeah, okay. But the ladies behind you, would you like to have a conversation? Yes? Yes, yeah, smiling teeth, yes. Yeah, so let's do that. Let's have a conversation later. So number 3, go.
Hi, I'm number 3, but people also call me Ava. So my question actually relates to the question number 2. So the same that we have to do like teaching, designing covers of journals and science communication. We have no time left for doing science. So my question is more like, do you think we should enforce science communication on everyone, or should we not more like stimulate more dedicated science communicators as a job?
Oh, okay. Specialization in science communicator. So you guys can hide in your caves.
Okay, so in short, I'd say if you mandate it, then lots of people won't do it very well. It's something that you have to want to do. But it needs to be encouraged, and it should be rewarded. So I think institutions should reward in the same way that they do other forms of, activity in the area of scientific research.
Because I have the feeling nowadays, like to do, get grants, you actually are enforced a little bit to do this. I'm actually started a science communication company myself some years ago, and we noticed that some of the audience don't want to be there,
yeah,
and some does. So that's why I'm wondering if it's working this way, if we should just really let the people that have affinity with it.
Just dedicate them to that, yeah.
I think mandating it is a bad idea, I do. But pointing out all the different kinds of things that can be part of that engagement umbrella can be quite useful for people, and encouraging scientists to think about this as a serious activity is also really important. But as soon as something is mandated, I think it loses its quality.
Yeah, it's scary, right? Let's just do a little poll. Audience, so the question is, do you think science communication can be mandated? Can people be dedicated to do science communication? Okay, can people, you all look very pensive, it's time to work. Okay, so those of you who think that there should be dedicated people, scientists to do communications, while the rest can hide in their labs, put your hands up.
I think that's a slightly different question, actually.
Yeah. I know.
Because I think the question is, should all scientists have to do public engagement? I think that's a different question from, is it useful to have people whose careers are public engagement? Absolutely, I think that is, because that provides a lot of the infrastructure around science communication. But I think there should be a space for scientists who are at the forefront of their fields to also be encouraged to engage.
Do that, yeah. Thank you so much for the correction. I stand corrected. So let's try that question again. Okay, so the question is, post the question, please.
So should all scientists be mandated to do some kind of public engagement?
Oh, that's an excellent catch, Alison. Yeah, very few. Again, can I see again? No, I won't ask you to come up to the stage, don't worry. Yeah, no one will do a poll. Oh, much fewer. Interesting, that's a good catch. Okay, thank you so much, number 3. Okay, we're going to get to number 4, and then I'm going to bring Alison to the audience. Yes, number 4.
Hello, I'm number 4, also known as Arpan. I'm from India, and I'm doing my PhD in developmental neuroscience, basically looking at how brains develop. So my question is how we can, what methodologies we can use to bring out the importance of people who are doing the science while communicating science. So let me communicate the question better. So when I ask some school students who is James Bond, they can kind of relate, okay, James Bond is this person. So, but when we talk about somebody's research, we just say XYZ et al. And they are doing this research. So how we can highlight their importance more and such that people can correlate that, okay, these are the people who are doing the science and we can appreciate their hurdles and their excitement behind the science.
So you're thinking about personalizing the science.
Personalizing, yeah.
Yeah, so there's David Attenboroughing the science. Something like that?
Yeah, yeah. How we can do that? Any idea?
Okay, and besides James Bond. Okay, thank you. Okay, how to get James Bonds? Anybody?
I mean, I think that the best way is the person who's done the experiment to present the science, which is happening here. I think that's, warts and all, I think that's the best way of doing it. I think it's a very different thing if you're presenting the science of someone else. So I think that in an ideal world, you would get these people to present their science, and that would be my answer.
Yeah. Valerio?
Well, within the scientific community, I think this happens when you go to conferences, right? And you really meet the people that have done that, including and especially the PhD students and the postdocs, right? Because usually, yes, the et al. Is the first author, right? But usually what remains in the mind of us is not the first author, right? Is that comes from the group of famous guy, right? Or the group of Zeilinger did that, the group of Aspect did that, the group of Gisin did that in my field. So in order to know that actually it was not the famous guy, right? But he's actually all the team, no? Maybe the best thing is to get to know them. And maybe when there is a media release or something, and that's what we do, for instance, also in CQT, you don't interview only the boss, right? You interview also, or maybe prominently, the people who did the legwork, right? I don't want to say that the boss is not important, it is, but also, I mean, that could be a way to do that.
Yeah, I think that's a really good suggestion, actually. And maybe that's a space for social media. So when the paper is published, you could have a social media clip that talks to the actual people that did the experiments and what their motivations were and what the challenges were. So the story, much more behind that research, because the human stories often get left out of papers, and I often think that's kind of to their detriment in a way.
Some journals do that. We had a Nature chemistry paper, and they interviewed exactly what you said, the first author, and asked her to write down, what the key things we discovered, what the problems were, and so that some of the good journals are doing that.
Yeah, it's a really good idea.
Yes, thanks so much, James Bond. Yes, and then, so we've done number 4. There is a question on social media, now that Alison, you brought that up. It's been voted by quite a lot of people. I'm just going to read it out, and then I'm going to do another audience poll before coming back to the panel. So it says, how do you think changing media consumption patterns affect the expectations of scientists and science comms today, e.g. the rise of popular experts on TikTok? So let me just do a quick audience poll. How many of you have communicated science in any way via any form of social media? It's decent, but it's not the majority of people. Can I see that again? How many have done that? Oh, interesting. Yeah, interesting. Yeah. Let me see. Yeah. So some of you have done that. Hi, sir. How are you? Where are you from?
I'm good. I'm from Sweden.
Yes. So what have you done on social media?
Yeah, I've done Twitter and LinkedIn. I also shared on Facebook. So I try to keep different levels on the different platforms.
Oh, very diverse. So what have you shared? Can you give us an example of what you shared?
It's been related to publications or also some stuff from other research groups that have done something really interesting for the field that I think others could see and benefit from.
And how often do you post on social media on science?
Not so often. Might be every month or so.
That's not bad. So every month, yeah? So those of you who actually post on social media, how many of you post more than once a month? So not very frequent, yeah. So you post more about your pictures looking at James Bond, yeah? And number 4. Okay, that's interesting. So I'm going to ask the panel. I won't do that for every poll question, yeah? Otherwise, no one will put up their hands. So I'm going to ask the panel, what are your thoughts about social media, TikTok, YouTube? Is anyone on Facebook still? Okay.
Yeah, okay, so my thought, well, I'm still on Facebook, yes, I'm not in TikTok, I'm not in Instagram, I'm not. I just joined Twitter because I was told that it's important to get postdocs, and that's the problem I want to bring up. However, I mean, we are here all agreeing that the communication of science is great and so on, but there is an element that I personally feel, maybe because it's me, is a bit backfiring towards the scientific community, in the sense that it gives rise to stars, or the people that are very prominent on Twitter, on social media, suddenly good scientists, absolutely good scientists, nothing against them, right? But they get a prominence, they get a voice, they get even within the scientific community, right? Suddenly what that guy says or that lady says, wow, no, he or she said that, no? And even my students or the students around us, when we say something different, it looks like us, who are you, loser, to contradict what the other fellows are saying, right? So this is a little bit of an unsettling situation. I find the social media personally very noisy in that sense. Yeah, but that being said, of course, there's no denying that it's very good to spread science there. Probably what the participant said before is, I mean, that's what I would advocate as well, right? Spread maybe your own results and also very good results of other people that you say, this is really something worth knowing. And the periodicity is not bad, huh? Once a month, I would say, you know, once a month, a big result in one field, yeah. How many do you want, right? One a day?
Alison?
Yeah, I do think that social media we're at the beginning of a big revolution in the way that science is communicated through social media. And it's a problem that it's not subject to peer review in the same way that the traditional publications are, but it certainly has a space. I find Twitter, for example, a useful way of keeping up with publications in my area. And it's because of the crowdsourcing involved, you can get quite a lot of new voices saying, oh, this paper is really cool, or I didn't like this paper because of that. So I kind of like that kind of democracy, because everyone can join in. But yeah, you have to also understand that the quality control isn't there either. So there's two sides of that kind of democracy. But you know, I can't really comment on TikTok or anything. My kids do that all the time, but I've never even been on. I'm scared of things like that. It's one of those things where I think when you set up an account, it's like you can't get rid of it somehow. It sort of starts to take over your life. And when I did do quite a lot of Twitter when I was involved in this TV programs, and someone, because I was made to, and then someone told me, if you're on Twitter and you don't post every day, you're kind of a loser. And I thought, well, that's just it's not going to work for me. So yeah, it's another thing to do as well.
Okay, thanks so much. Okay, I'm going to ask 5, 6, 7 to step up to the microphone. 5, 6, and 7. Yeah, so we're going to take 3 questions together. Who's 7? Who's 6? Yeah. Huh? Oh, it's covered. Okay, thank you, ma'am. Thank you so much, yeah. Okay, so can I have 5, 6, and... Okay, I don't know what number you were, but 5, 6, 7, 8. 8.
Okay.
Thank you. Yeah, so can I have 5 questions, 6's question, and then... I don't know what you are. 8's question. Okay, let's take them together. Okay.
Yep. Hello, I'm Ryan. I'm just out of high school, actually. I'm from Singapore. And yeah, thank you so much for your sharings. Yesterday, actually, one thing that came up during one of the panel discussions was that in relationships, it's very important to think, how am I relevant to the other party? So in a similar vein, in communication, when thinking of how would I make this relevant to my audience, what goes through your mind? For example, even for Dr. Valerio, teaching quantum physics to someone who's studying Chinese culture, for example. Thank you.
How to make it relevant, yes. And then can I have number... Ma'am, were you 6?
Yes.
Thank you.
Okay.
Yes.
So I am Zainab. I'm from UCSI University from Malaysia. I would like to ask actually a few questions, but I'll try to make it into one question. So in each era of any study or subject, there is some type of characteristics that a science communication specialist could have to be a great one. So my question is, in 5 years ahead of us, what are the special characteristics that we should obtain now to be able to be a great science communication specialist? And my second question is emotional intelligence or sociology important in science communication? Because we need to understand the audience. A lot of us have the knowledge. We have the knowledge to say it, but some of us do not have the aspect to communicate or to understand when the audience are feeling bored or when the audience need a certain information. So how do I know this? And also, when can I call myself a science communication specialist? Because I do give some talks from now and then through online platforms like Zoom, Teams, but I want to know, when can I call myself a science communication specialist? Thank you.
I can say that right now. You can call yourself a science communicator right now. Yeah, you're communicating very well. Thank you, ma'am. And sorry, 8.
Hi, I'm Taya. I'm an astronomer if I want to communicate, and a high-energy astrophysics if I don't. I'm asking in the context of communicating with science skeptics, which I find very challenging, and especially one point is we often say, well, these are the facts, you can see the facts. And as far as I can tell, at least for me, I can't see the facts if it's biology. I can't even see the facts if it's another field in physics. The only field where things are rigorously based on facts and proofs is maybe mathematics. Even in my own field, I kind of believe previous results and trust science to correct itself. And I'd be interested in hearing how those nuances are communicated, because I'm asking, when I say believe the facts, I'm asking someone to trust the scientific institution, the scientific method, more than actually understand the basic facts.
Thanks, that's an excellent question. I'm going to ask the panelists to hold on to answering that question, because there's a bunch of questions on Slido that's linked to that.
Okay.
So I'm going to read them later after we've addressed the questions for 5 and 6. Yeah, so just hold on to that. So the question of 5 and 6, just to recap, is about being a relevant communicator, very much like having relationships. Yeah, I presume you're having one right now? No, okay, you should have one. And then also about emotional intelligence, and I don't know about the one about being labeled a specialist, but we can get to that too, yeah. So, and also the idea of the characteristics, that's a very specific question, of being a communicator in 5 years. So there's a whole bunch of things about being relevant and predicting the future.
Well, I mean, since you asked me, you mentioned my case, right? I mean, relevance is a relative concept, right? So I find that sometimes even the research I'm doing, because I'm a very kind of fundamental theorist, right? Well, one of my big problems writing grants is when it comes to the line, what is relevant for Singapore? Right? We know the problems of Singapore, water, nothing, okay? Well, so the relevance for Singapore of my research is forming people, educating people, forming scientific mentality and so on, right? Is it relevant? Well, I don't know. I mean, as in, will the grantor give me money for that? Similarly for outreach, I mean, you can't expect that, I can't expect, maybe you can, depending on what you speak about, that the whole population will be excited about what you are doing, right? And in my case, for instance, I know that the whole population will not get excited by what I'm doing, but maybe those that are more philosophically oriented, more what is nature trying to tell us and whatever, these people might catch. So I think, of course, you need to be relevant at least to some people, right? So that's the thing. So you have to identify the group of people that would listen to you.
Yes, and then, yes, Alison, and then maybe Sir David as well.
Yep. Yeah, so I do think if you can find out about the audience, then do. So I think sometimes organisers of talks and things like that are not really very good at telling the speaker who the audience are, why they're going to be there, How old are they? How many of them are likely to come? All of these questions are really important because that allows you to prepare. And I like the way that you use the word emotional intelligence, actually. And I've never really thought of it like that before, but I think it's true that, you know, to form you form a kind of empathy and a connection with your audience, then it's really important. You need to think about why are you here? Where have you come from? Why are you here? What do you want? What do you want to gain from this talk? And that all kind of helps with the pitching it right. How do you know when people are not following you? Actually, I think it's really obvious, because they start using their phone, or the lid of the computer comes up, or sometimes they fall asleep, or sometimes they just leave. So I think it's a bit terrifying. You think, oh, I'm losing them. I'm definitely losing them. And you have to try and get them back and round them up again. And, you know, so sometimes you do need to do an in-talk adjustment, or give people space to say, I haven't got a clue what you're talking about.
What do you think? Then have you got them?
I got most of them.
Yeah, I think you got most of them. Yeah. I have to
but
say,
this is a very self-selecting audience.
It is, and I'm very surprised. I've seen very few of you using your phones. Is this a?
They might be just tweeting it though.
Yeah, they might, but they're not even doing that. So most people pretend they're tweeting, but they're actually checking their social media accounts. But no one's doing that. You guys are brilliant. Give yourself a big round of applause. Yeah, very, very committed audience. Yeah. I did spot one person yawning though, who was on my left. Yeah, there was somebody who yawned right at the start, but because he stopped yawning, I have forgiven him. There's someone leaving. Yeah, leaving.
I'm not just capturing for social.
Oh, I'm sorry. Okay, well done. That's what you say when you're leaving. I'm capturing photos. Okay, Sir Dave, can you predict what will happen in 5 years?
Predicting in what
sense? I'm sorry. Sorry to put you on the spot. Just say what you wanted to say, and then we'll talk about predictions.
So, I mean, certainly from my point of view, the relevance is quite straightforward because we deal with problems that people can immediately identify. So I really haven't faced the issues with relevance. But I would sort of frame it slightly differently. I think the talks are more you articulating why you're interested in your field and why you enjoy it, and trying to bring the audience along with you rather than trying to say, you know, it's going to cure cancer in 10 years. I think I would frame it as I'm trying to advertise why you should be a scientist, why you should study chemistry, and this is what excites me, and just try and share your enthusiasm and your experience with the audience. And it's less important about the relevance, it's more about the enthusiasm and explaining exactly what drives you to get up every morning and go and do your job as a scientist or a chemist.
I like that very much. It's almost like personalizing that experience for you. Yeah, and it's not about telling them what they should or should not do. Shall we all pose for a picture? Yeah, she's actually taking photos, yeah. Thank you so much. Yeah, thank you so much. Yeah, you can leave if you really want to, because you just taking the photos, right? Yeah.
But I just add to that. I think my limited experience is that the talks are better if you say it exactly, make it much more personal. I was excited when we saw this. I mean, I started putting photographs of myself in the talks just to make it much more of a personal story than a bunch of facts that you're explaining to people.
And I've seen how you open your other talk here. Yeah, and I love how you drop things like loops and luck and how colorful your labs are. So those things actually make you less intimidating. Yeah, because when I read your biodata, I was intimidated. Yeah, but okay, enough about me. Okay, I'm just going to address that fantastic question for number 8. Are you still there? Number 8, yes, number 8, that was a superb question. Number 5, you may sit down.
Yeah, thank you.
It's all right, and start reflecting on relationships. Okay, so number 8, that was a superb question. Like, how do you know the facts if you are not in the industry? I'm going to read off a couple of questions that's linked to that, if you don't mind, yeah. So, panelists, I'm going to read off several questions on trust, science, and fake information. Okay, so this is one bit. So how can media promote a culture of responsibility in science reporting? And what steps can be taken to address inaccuracies or misinformation when they occur? This is from Mr. V. Vijendran. Then there's another question on how do you increase trust in science within communities which have been adversely impacted by scientific research and the scientific academic system. Okay, there are a few more. I'm sorry to bunch them up, those are at home, but there are too many questions on there, and they're really good. Could you recommend how to hold on to the situation that fake science comes up from influencers not specializing in that field, but influencing society? There are a few more on fake information and trust. Okay, so there's a question on how do scientists, science communicators, and journalists work better together to fight disinformation? And there's a final one on trust, which is, on the note of experts on social media, is it our responsibility as real scientists to call out fake scientists, to refute fake science? So it's a whole bunch of questions on trust, on facts, on media, on representation, and social media. Sorry about that. There's a whole bunch of questions. Choose to address any part of that.
I can start. Thank you for your very clear statement and question. I really enjoyed listening to that. And I was interested in that you talked about skeptics. And actually, skepticism is a kind of origin of science in many ways, because the skeptics were the people that tried to apply more logical explanations for things that were kind of very mysterious. So kind of skepticism sounds like it ought to be a good thing. But now, of course, we have conspiracy theories, which is a different thing, I think, and something that is very hard to address and hard to engage with. But I do think that just shouting the facts doesn't really help. And finding out why people are in that conspiracy theory. What is it about their culture, their background, their belief system, their experience that means that they want to reject the scientific explanation? And I think that's the key. And I don't know what the answer is, but I think that talking to these people, I'm not saying that that's something that we can all do as scientists, but I mean, maybe the social scientists are the ones to start to do that for us, to try and find out more about why people hold these views. But I do think that trying to understand where the views come from is important.
This is just shouting the facts, like you said, yeah.
But correcting, trying to correct the record, I think that's also really important as well. And it's relentless. I've got colleagues who are climate scientists that have to relentlessly put out information about human activity and climate change to counteract the narrative, because that's hijacked by politics. And then it becomes very important to try to put out correct facts that counteract wrong facts. Fake facts. But conspiracy theories are very difficult to deal with.
Sorry, I'm just trying to wrap my head around fake facts. Yeah. Alternative what? Alternative facts? Facts, yeah. Right, yeah. Just before we get into the other parts of that discussion, is it important then to stay close to politicians?
It's a task for some people. I mean, I don't think that every scientist should. It's important that some scientists. I mean, science is not. There are people that are in the lab the whole day, all the way to the people that go lobbying the whole day, right? And then in between, there are all kind of spectrum. So, I think that some people have to do that. I mean, we scientists cannot just say we ignore politics, but at the same time, some of us can also say, well, it's not my task.
Are you close to any politicians?
In Singapore?
Yes. No, you don't have to name them.
No, I'm not.
You don't have to name them.
I'm not. No, I don't know any. Yeah, I've not been in any political role till now.
Would you like to be introduced?
Why not?
Okay, thank you. Sorry, David.
So I think that science communication is also to the politicians, because certainly in the UK, very few of any of the MPs actually have a degree in science. And I think you are communicating to the politicians and the rest of society. Some people need to make the case for why you should be doing science and how it can contribute to society and how it can improve human health, etc., etc. And so I think it is important to not directly speak to the politicians. And certainly in the UK, we've had politicians come round who are very interested in learning more from the scientists. So I think some politicians are also very engaged, but I think it's very important. We need to be making a strong case for science funding, science teaching, and communicating with the politicians is part of that.
Do you think it's important for scientists to be politicians?
There aren't many scientists who are politicians.
There aren't, yeah.
There are not. There was one student I jointly supervised in Cambridge who became an MP for Cambridge, but it's very, very rare. I think I think it would be good if that happened, but it's certainly in the UK, scientists do science and people who do PPE at Oxford are politicians.
Yes, PPE. Okay, quick poll question. How many of you think it's important for scientists to be friends with politicians? Okay, hands down. How many of you would like to be friends with politicians? Very few. Yeah, strange, right? Yeah, strange. Okay, so one last question related to that, and I think that was a very important question, and I think Alison has also talked about that. How do you fight misinformation? Do you think it's important for scientists to take a very active role in that?
Well, first of all, maybe to frame my answer, right? I have a very Bayesian view of science, which may be very scientific as well, which is that evidence leads you to update your beliefs, right? Evidence does not trump your beliefs. So maybe this is an answer to the question that as it was phrased. So, and it also addresses conspiracy theories. If someone has, I mean, if you do some Bayesian analysis, it's very simple, right? If someone believes, for instance, that they cannot get COVID, well, whatever happens, they have not gotten COVID, right? Because their prior is 1. And you cannot trump a prior of 1, but you can trump a prior of 0.99 with sufficient evidence. So sometimes the problem is this, right? Sometimes the problem is that you have to present more and more evidence, right? We cannot, as Alison has just said, we cannot just go there and say, hey, I am telling you the truth. Forget about all that you know. I'm a scientist, you know, the priest of the truth. We can present more and more evidence and hope that the people, their priors are not so biased that after enough evidence, they realign maybe a bit better. That would be my solution to that.
Sure.
Of course, from time to time you may have to come out and say, no, this is nonsense explicitly, no, to some things and so on. Those who are more active in the social media do it. Yes.
Yeah, yeah. You say
and,
really,
I think they should do it. Those who really want to be active in the social media, they should do that.
Just give it straight, like this is bull. Yeah, this is bull. Yeah. Okay.
I think it's also difficult because there's some experiments where you don't get a definitive answer. So this was a big problem with the pandemic, and communicating that. It's not black and white, it's some scale of grey.
Yeah.
And I think it would be better if generally the public were better educated in science and better understood how science operates. And I think there's a general need to have a more scientific understanding public.
Yeah. So versus just facts 1, 2, 3. Yeah, it's very strange. What's that thing? People who do not know enough tend to be more confident than those who do. Yeah, that's interesting. I don't know who said that. Yeah, probably nobody said that. Okay, right. Can I have 9, 10, 11, please? Are you still here? Yeah, okay, 9, 10, 11, still here. Thanks. So who's 9? Sir,
9.
you're 9? Yeah. Oh, you're on stage. Yes. You're on stage. Yeah, earlier?
Yes.
Yeah, okay.
I had a presentation.
Well done, you're communicating. Okay, so can I have 9, 10, and then 11? So 9 first?
Yes, I'm Qasim, Mr. Qasim from Pakistan. My question is related to dual-use research of concern, being a biosecurity fellow. So I wanted to ask whether, considering the dual-use research of concern, whether science must be communicated openly or not.
Oh, okay. Because especially in the midst of research, yeah.
In the midst of research, and actually some of the research may be misused, so This is a controversial topic, but just wanted to know your views.
Fascinating. Thank you. And number 10? Yes, sir.
Hi, everyone. I'm Isaac Nabe from South Africa, and I'm also from the quantum optics community, and I'm such a fan of your work, Professor. So one of the things that I realized when I started entering the field is, our supervisors give us stacks of review papers, and you happen to write a lot of beautiful review papers, and that's also a form of science communication to scientists. And usually review papers sort of set the scene for the field, give some of the open questions, and even should be informative. And I'd like to ask if there's other tools that can be used to communicate science more effectively to actual scientists, so we inspire more people to actually enter these fields.
Interesting, yeah. So tools for scientists, openness in sharing research to avoid misuse or whether it will be misused. And then number 11, ma'am.
Hi, I'm Danica. So I'd like to ask, how do we know if we're doing enough? I think a good example that I have is the Bloomsbury Festival. So for those who don't know, this is a festival that happens in London where research groups are encouraged to create these interactive exhibits for the public. It took quite a lot of effort from our research group to create an exhibit, but majority of the population who attend or participated are like children from 7 to 12 years old, or just some adults not in the field of science who just want to pass their time in London. So at the end of the exhibit, we started to question how productive was it actually. I'd like to think that we've touched some young minds back there. I wouldn't want to in any way discredit the Bloomsbury Festival, but it would be good that if we could perhaps assess where we would be putting our efforts in and how we can criticize how well we're doing. Perhaps a key performance indicator, apart from, let's say, the number of followers we have on Twitter or listeners on Spotify. How can we tell if we're doing enough engagement?
Thank you, ma'am 11. Can I ask you a question back? So for your, you look terrified. Okay, I'm so sorry. Okay, so for your participation in the festival, what would you have seen as success? Apparently, 7 to 12-year-olds are not an indicator of success.
It's not that, but perhaps what we do in our research group is a bit new, and it's not really something that's existing in the field. Maybe one thing that I'd be happy to see was the spark in the kids' eyes, saying that, oh wow, science is incredible. Maybe that was a performance indicator for me, but
okay.
I wouldn't be able to measure that.
So spark in their eyes, yeah. So they all had dead eyes.
Dead eyes?
Really, yeah. Okay.
I hope not.
Don't worry about them. They'll never be scientists, yeah? Yeah, they'll never be scientists. They'll be politicians. Okay. So there are 3 questions. Thank you so much. So the 3 questions are, just let me do a quick recap. So something about sharing of science and research with the potential and the danger it could be misused. There's a questions on, are there other tools besides review of papers to communicate to scientists and future scientists? And the third one is, I'm so sorry to reduce this to this, but it's really like the KPI, the measurement of success. Are we doing enough? So feel free to answer any of these 3 questions. We have about 7 minutes left.
Yeah. I will take the half of the first and the second, right? So, I mean, science can be misused. I don't know what you were referring to. I don't know where is seated. If you are referring to misuse in the sense that with nuclear fission you can create an atomic bomb, or whether your competitors may, if you are too open. So in my case, what can happen is not the atomic bomb thing, but it's more like if I go around and speak too open about my research, maybe some colleagues of mine who go to a conference and so on, they hear the thing, and they start also working on that and scoop my results because maybe they work faster than. In that case, my suggestion is, after trying all kind of things in my career, being open, you never regret. At worst, you lose a paper. At worst. But then after that, people know that you are an open person, that you can you speak about it. And by the way, if really is dishonest what the others have done, sometimes it's not, it's just sheer competition. If it's really dishonest, that you told them something, they said, oh, okay, well, and then following day they publish a paper, then you know with whom not to talk anymore, right? But at the start, I would give trust to everybody, rather than start suspecting that maybe some here in the audience are trying to steal my things, right? So that would be my answer. Concerning the other question about the review papers, I think another way to educate them is not, maybe it's kind of obvious, but give credit where it's due in your papers. And nowadays, in my field, in our field, I see all kind of young people citing, oh, this is a very exciting topic, references 2020, 2021, 2020. This was done in the year 2000. And some people had the idea in 1970. But no, it looks like it's only my supervisor and their friends that have just done the latest work, and now I'm providing that. And unfortunately, this creates publications, right? This creates a cycle. If you say that it was already done in the 1970s, the prestigious journals tells you, oh, it's no longer actual, right? So there is a danger there we have to play with. I cannot continue anymore. I let the ground to my other colleagues, but I think the more is clear about the introduction, what you have done, what you have not done, what are the assumptions, what are not the assumptions, it does help in the long term.
Thank you so much. Yeah, and that's about the open-mindedness that Alison was talking about. Yeah,
I was just going to completely agree with Valerio about open communication, and I think the advantages nearly always outweigh the disadvantages. And that, you know, you're much more likely to find someone in the audience that's going to come up to you in the end and said, "Oh, I was really interested in that. It fits with something we've been doing, and maybe we could collaborate." That's where really important discovery often happens. So, and yeah, and if you lose a paper, the grand scheme of things, it's not the end of the world. And you do, and then maybe there's a way of letting the community know that that happened. Yeah.
That sort of answers the question as well. If someone comes up, it's not about numbers to say that your paper resonated with me. I would like to collaborate with you. Yeah, that's actually a huge one. So I should be seeing a lot of people coming up later at the end of this talk. Okay, man. Sorry. Oh, I broke your train of thought. Yeah.
It's very easy to break my trains of thought. Yeah, but that was about open communication.
Thanks, Alison. So, David?
I agree entirely. I don't think I have much to add. I would just be open and behave correctly, and I think that's the best way to behave.
So, David, what's your measurement of success?
There are lots of measurements of success. I think in science communication,
yes.
I think that ultimately, it becomes a question of why you're trying to communicate the science. And I think only if you define what you're trying to do. So if it's the kids smiling, you know, then you count how many kids smiled, and that's your measurement of success. If you're trying to encourage people to do science, then you have to measure how many people did science. So I think certainly my take on it, the science communication, is you really need to think what you're trying to communicate.
Absolutely, yeah. So it's the objective. It's not one single indicator. Excellent. How about you, Valerio? What's your measurement of success?
Well, I try not to think too much in these terms. Of course, at some point, I agree with you that you
book sales.
Well, no, that would not be a very good measure for myself. But anyway, now I tend to agree with you. Sometimes we do something and then we spend a lot of effort doing that, and then it seems that this effort was wasted. I think if you look in the grand scheme of humanity, right, most of us do a lot of things that don't seem to have a great impact, right? But at the end of the day, that's why humanity goes on. I mean, this sounds very, very, flying very high, right? But we cannot always do just the thing that is needed to get the efficiency back, right? There are moments where you do things, you spend some effort preparing this exhibit. Not many people came. Well, so be it, no? I spent some effort preparing my soccer game and I fell jogging last week and I cannot go, right? I mean, well, that's it, right? I mean, so, sometimes it's not a matter of efficiency. Sometimes where we do things, and that humanity just goes because we do things.
And it's not instant. It's not like,
instantaneous.
It's not like social media or TikTok. Yeah. It's not instant. You get followers, you get likes.
Or maybe there's never. Maybe that thing will just be forgotten. But
or maybe in 10 years'
we enjoyed
time,
and maybe preparing that activity actually taught you something about your own research. Maybe you got some deeper insight by thinking about that in a different way.
Or maybe you inspired someone who became a scientist and solved the climate crisis, so you never know.
In 5 years' time, who is asking for predictions? Let me get a measurement of success for us today. Are you ready for this? Shall we get the measurement of success? Okay, let's do a measurable success. Yes, yes, let's do it. Let's do it. Yeah, let's do it, girl. Okay, so how many of you think that this panel today was effective? Was effective in no? Oh, I'm not finished. Okay, okay. Was effective? Okay. How many of you think it was effective? I know, but he stopped me. Yeah, so I had no idea. I couldn't finish. Yeah, effective. Okay, so I had many. This was effective. Yeah, in telling you what you can do about science communication. Okay, now vote. Interesting. Interesting. Yes, yes, yes. And okay, I have 13 seconds.
Could I be like 12? So I'm postdoc from...
Just to say, 12 as in...
Number 12.
Not 12 years old.
No,
so
no.
that'd be like 12 because of that 12 thing. Yes, yes, ma'am.
I'm Kamila Kokabi. I'm a postdoc from Nazarbayev University in Kazakhstan. So discussion was very productive. So during my study, I'm involved also in teaching. When I'm encouraging students, new generation for reference for the study like 19 something, they are mostly saying, do you have something new, fresh, like review? So I would recommend for the like old generation, let's say, I don't know how to express, like to write for your research new updated review. So it would be benefit, like it would be cited also again your research with like updated.
So updating your research, keeping it fresh for a new audience. That's a very good way to end today. You don't always have to invent new things, but sometimes the things that you've done can have multiple bites. Multiple bites. Thank you so much, Camilla. Right? Yeah, thank you so much. Thanks for being 12. Okay, so with that, I would like everyone to give the wonderful panelists today a huge round of applause. Okay, and thank you so much. You guys have been a very good audience, and I think we should say thank you to all of you. Okay, so have fun for the rest of the summit. Thank you.
Thank you, Mr. Tan and members of the panel for the engaging discussion and insightful comments. We will now be taking a break. Before you go, I wish to share that this morning's online engagement is Quantum Ask Me Anything by the Center for Quantum Technologies. So please join the Give Me platform for the online engagement. And for the participants of the poster sessions, if you have yet to collect your certificates, gentle reminder to collect them from the registration counter during the break. Otherwise, you may proceed for the refreshments at the usual place outside, and please be back here by 10:55. Thank you. Welcome back. We continue this morning's program with Prof Thomas Südhof. So Prof Südhof will be joining us virtually for his talk. His talk is titled Towards Understanding Synapse Formation in Health and Disease, Implications for Neurodegeneration. The Q&A session will be moderated by Prof Lim Ka Leong. Prof Südhof, please.
All hear me? I wish I could have been there in Singapore in person. It would have been so much more enjoyable, I hope, for everybody, certainly for me. I miss visiting Singapore, where I've been several times before the COVID epidemic. And I look forward to my next visit to this wonderful country. What I want to do today is indicated in the title of my talk, except that when I gave this title, I didn't realize that I only had a short time to speak. And so I need to now change that title. And I will change it to reflect what I'm going to talk about, which is a scientific project that we are currently doing in the lab and that I find particularly interesting, that relates to disease, but I'm not going to have time to tell you the details about this disease. In fact, I want to specifically talk only about a very, very brief introduction to one big area of research, which is synapse formation. We all know that the brain functions in processing information via neural circuits. What you see here is a schematic of a neural circuit, a microcircuit, where 3 neurons are connected to each other by synapses. So circuits are formed by synapses. And the circuits process information by transferring and changing the information from one neuron to the next at the synapses. The synapses are the fundamental computational unit of the brain that exhibit an amazing speed, precision, and plasticity. There are many properties of circuits that are important for information processing. And one of them, possibly the most important, is the location, identity, and properties of the synapses. What my lab is trying to do is to understand what molecular rules govern the formation and specification of synapses and what happens when these rules and the synapses are disturbed in disease. Our fundamental proposition is that in a synapse, as shown here in this electron micrograph, 2 cells, 2 nerve cells usually, form an intercellular junction. And that junction is specialized for transferring and processing information. The idea is that this junction is controlled by transsynaptic adhesion complexes that mediated bidirectional signaling, thereby telling each neuron about their identity and instructing each neurons with regards to the plasticity, release probability, and other properties of the synapses. Many candidate synaptic adhesion molecules have been described that are seen schematically in this slide here. This is not an exhaustive presentation. There's many more, but these arguably are the most important. What you see here on the left side are the presynaptic terminal molecules, on the right side, the postsynaptic. And you can see that on the left side, there are several hub molecules, such as neurexins, that interact with lots of postsynaptic molecules. Although we try to understand all of these molecules, in the end, there's only a very superficial understanding at present. And what I want to do is focus on one particular set of molecules that I'm going to discuss today. And what I want to try to illustrate to you today is how we can use molecular approaches to dissect the molecular logic that governs the transsynaptic specification of synapses. And specifically, what I want to focus on today is neurexins, which are presynaptic molecules that interact, as you can see here, with a wide spectrum of postsynaptic ligands. In fact, more than 50 genes encode ligands for neurexins. I don't want to give you a total view of this family of proteins because the time isn't sufficient. I only have 30 minutes. I only want to tell you about one particular project within that bigger project, which is again part of an even bigger project, namely to understand the molecular logic that dictates synapse formation and specification. So this is what I'm going to tell you about today. I'm going to tell you first about the physiological function, not of neurexins as a whole, but of one particular event in neurexins, namely in neurexin alternative splicing. And then I'm going to tell you about the amazing mechanism that is used to actually mediate this function. So let me start at the beginning. And let me first introduce you in more detail to the presynaptic adhesion molecules called neurexins. What you see here schematically is that these proteins come in two principal flavors, alpha neurexins and beta neurexins. The alpha neurexins are larger versions with many more domains than the beta neurexins, which are shorter versions. Alpha and beta neurexins are encoded by 3 genes, each of which has independent promoters for alpha and beta neurexins. They're localized to synapses, at least in part. Interestingly, thousands of mutations were identified in neurexins in autism, schizophrenia, Tourette syndrome, neurodevelopmental delay, all kinds of neuropsychiatric disorders. And we have, in fact, in studies that I don't have time to discuss today, analyzed in neurons derived from human stem cells and from patients the effect of such mutations, demonstrating that they do impair synapses, which I'm not going to discuss because of limits of time. What struck us when we first identified neurexins was that they have a tremendous degree of alternative splicing. They're among the most alternatively spliced molecules in biology, creating thousands of isoforms that have been validated by PacBio sequencing. And what I'm going to talk about today is one particular splice site called SS4 that is shared by alpha and beta neurexins as shown here. And finally, as you probably know, we have already, as I already mentioned, oops, doesn't work, excuse me. As I already mentioned, neurexins have many, many ligands. So what I'm going to talk to you about today is, in fact, the isolation, the role of one particular splice site in neurexin splice site 4, a very limited, very restricted question. And I'm going to discuss this with you today as a way to illustrate to you how we try to address this big question by going into detail in one particular molecule, in one particular splice variant, marching from one molecule to the next. And so the starting question of the project that I'm going to discuss with you today is, what does alternative splicing of neurexin at SS4 actually do? And so to address this question, we used a genetic approach. We made mice in which alternative splicing of neurexin 3 is under control of Cre recombinases. These mice were made in such a way that in the knock-in, as this is, without a Cre recombinase, you always get a splice variant with SS4 in. As soon as you add recombinase, you always get SS4 minus. And this way we can control very tightly the splice variant of neurexin 3 that's being expressed. Using this approach, we then analyzed hippocampal neurons in culture from these mice electrophysiologically. What you see here is that when you do this, the neurexin 3 SS4 plus has a decrease in synaptic strength mediated by AMPA-type glutamate receptors, whereas synaptic strength mediated by NMDA receptors is normal, as is the synaptic strength of inhibitory synapses. So this was a surprise because this is a postsynaptic phenotype, and it raised a number of questions that we needed to address. Namely, one is, being a postsynaptic phenotype, is this really a presynaptic effect? Does it act presynaptically, transsynaptically on a postsynaptic neuron? And so in order to address this question, we used an in vivo approach that is illustrated here. We injected stereotactically into the CA1 region of these mice, of the knock-in mice, AAVs that encode either wild-type Cre recombinase or a mutant control Cre recombinase at P21. And then we analyzed these mice by electrophysiology 2 weeks later, recording from the subiculum neurons and stimulating the axons that are formed by CA1 neurons and form synapses on the subiculum neurons. This preparation allows a precise analysis of a presynaptic effect because only presynaptic CA1 neurons are infected. The postsynaptic CA1 neurons are not infected with AAVs, as you can see here in the green color that is created by the AAV infection. And when we did this, we observed the same phenotype as in cultured neurons. We found that the neurexin 3 SS4 plus knock-in caused a decrease in AMPA receptor-mediated glutamate receptor responses, whereas the presynaptic excision of SS4 reversed this phenotype and made it appear to be the same as in wild type. We detected no effect on NMDA receptor-mediated responses. So this result confirmed that presynaptic neurexin 3 alternative splicing at SS4 controls postsynaptic AMPA receptor-mediated responses. And further studies showed that this operates at the level of the receptors themselves. So this constitutes a mechanism by which a presynaptic neuron can tell a postsynaptic neuron how to behave, what properties to have. But it also raised questions. The neurexins are highly homologous. Do the other neurexins perform similar functions? Or do they do something different despite the fact that they're so similar? And so to address this question, we generated and analyzed the same type of mutant mice for neurexins 1 and 2. And when we did this, we got a big surprise in that the neurexin 1 mutant had no effect on AMPA receptor-mediated endocytosis, AMPA receptor-mediated responses. They were exactly the same. But when we now analyzed NMDA receptor-mediated responses, we saw a dramatic increase in synaptic strength. So different from the neurexin 3, where there is a dramatic decrease in AMPA-mediated responses, here there is a dramatic increase in NMDA receptor-mediated responses, suggesting that the neurexin 1 and neurexin 3 have completely different functions affecting glutamate receptors at the synapse by a transsynaptic mechanism, despite the fact that they are so similar. And finally, neurexin 2 SS4 mutants, to top it off, had no phenotype. They didn't affect either the AMPA or the NMDA receptor responses. So the three neurexins are intrinsically different. And these experiments, together with additional experiments that I don't have time to discuss, led to the conclusion, as shown here, that neurexin 1 SS4 plus upregulates NMDA receptors, whereas the neurexin 3 SS4 plus downregulates AMPA receptors, which represents a mechanism of transsynaptic control contributing to synapse diversity. Presynaptic neurexin 1 and neurexin 3 alternative splicing by this mechanism dictate the postsynaptic glutamate receptor content that is characteristic for any given type of synapse. So you might ask, I hope, how does this work? How is it possible for two different neurexins that are so similar to cause the regulation transsynaptically of two different types of glutamate receptors? To address this question, we have to go back to this diagram which illustrates the ligands of neurexins. And it turns out that there's only one type of ligand that binds exclusively to SS4 plus splice variants of neurexins. And these are cerebellins that are secreted adapter proteins. And these cerebellins come into two different types, 1, 2, 3, and 4, that bind to different postsynaptic receptors. When we analyze cerebellin expression in the subiculum, our target area, we found that only cerebellin 2 is expressed at very high levels, and therefore we used cerebellin 2 knockout mice to test whether they might be responsible, whether they might show a similar phenotype. Now, cerebellins have traditionally been implicated in synapse formation. So we first measured synapse numbers in the cerebellin 2 knockout mice in the subiculum and found no phenotype, no change in synapse density. We then measured synaptic transmission, and gratifyingly, we observed both an increase in AMPA receptor responses and a decrease in NMDA receptor responses, as shown here. So what this means is that the cerebellin 2 knockout represents a mirror image of the combination of neurexin 1 SS4 plus and neurexin 3 SS4 plus effects. Neurexin 1 SS4 plus increases in NMDA receptors. If you take away cerebellin in a wild type, you get a decrease, presumably because the endogenous neurexin 1 signal is no longer there. Neurexin 3 SS4 plus suppresses AMPA receptors. If you take away cerebellin, you get an increase of AMPA receptors. Again, presumably the endogenous neurexin 3 signal is no longer there, suggesting that strikingly and unexpectedly, cerebellin 2 mediates or transduces the effect of both neurexin 1 and neurexin 3. So what is the cerebellin 2 receptor on the postsynaptic site? Now it turns out that only one family of proteins has been shown to bind cerebellin 2 postsynaptically, and that is a family of protein called the GLUD1 and GLUD2. And these are remarkable because GLUD1 and GLUD2 are actually homologs of AMPA and NMDA receptors, which are also homologous to each other. So we tested whether GLUD1 or GLUD2 might be the target of this manipulation. We deleted GLUD1 using CRISPR approaches. And as you can see here in culture again, with GLUD1 knockouts, there is no change in synapse numbers. But when you measure synaptic function, you get a phenocopy of the cerebellin 2 deletion, in that there's an increase in AMPA receptor responses and a decrease in NMDA receptor responses, suggesting that the cerebellin 2 effect is mediated via GLUD1. And in vivo experiments showed the same results. Moreover, double mutants demonstrated that GLUD1 functions downstream of neurexins and cerebellins, suggesting that this following pathway operates, whereby neurexin 1 and neurexin 3, SS4 plus, regulate differentially NMDA and AMPA receptors in opposite directions. And this effect is mediated by the same transduction machinery on the postsynaptic side, raising the important question of how on earth can the same postsynaptic GLUD1 molecule transduce distinct neurexin sequences, distinct neurexin signals. And so the hypothesis that we came up with is that GLUD1 transduces distinct neurexin signals via different cytoplasmic effectors, that it basically differentially has different modules for the different neurexins to mediate their effects. And to test this, we probed effects of mutations in the cytoplasmic GLUD1 sequences. We identified 5 conserved sequences in the cytoplasmic tails of GLUD1, and we mutated these 5 sequences, which are shown schematically here, to exactly the same target sequence, except for the C-terminal PDZ domain binding motif, where we exchanged it for a different type. And then we tested rescue. And what you see here on the left is the AMPA receptor-mediated responses. You can see that the knockout, as shown before, dramatically increases the responses. I've shown you this before. What you can also see is that only 1 out of all these mutants did not rescue this phenotype, suggesting that there was one particular sequence that was essential for the AMPA receptor-mediated regulation by GLUD1. When we did the same experiment for the NMDA receptor-mediated responses, you can again see the suppression by the knockout, and you can see that again, only 1 mutant did not rescue. The other ones all rescued. But the mutant that rescued the NMDA receptor-mediated response was different from the one that rescued the AMPA receptor-mediated responses, demonstrating that it's different sequences in GLUD1 that mediated the 2 different regulatory effects. In other words, the mechanisms are, as you would expect, different for AMPA and NMDA receptors, leading us to this model here, whereby the GLUD1 cerebellin 2 complex regulates NMDA receptors on the one hand and AMPA receptors on the other hand, depending on whether it's presynaptically neurexin 3 or neurexin 1. Now, in my final sets of experiments, I want to explore further how this might work. As I mentioned already, GLUD1 is structurally similar to AMPA and NMDA receptors, which operate by a mechanism whereby ligand binding to the extracellular sequences causes a conformational change in the transmembrane regions that then opens an ion channel. So the question then arises whether GLUD1 operates by a similar conformational change in the transmembrane domain like AMPA and NMDA receptors, whether that is conserved or whether they have a different mechanism. And so to test this, we simply took away all transmembrane regions and, in fact, the transduction domain as well. And we made a simple variant of GLUD1 that is shown here schematically, where we change the transmembrane regions, the 3 transmembrane regions, and convert them into a single transmembrane region that we get from CD4. We also took away one of the extracellular domains so that the only sequences that we have are the N-terminal cerebellum binding sequence and the C-terminal cytoplasmic tail. And then we measured rescue. And by the way, we also did the same thing for GLUD2, the homolog. And what you can see here is that these minimal GLUDs rescue both the AMPA receptor responses and the NMDA receptor responses. So this means that GLUD1 and GLUD2 have the same functions and operate not by any conformational transmembrane region change, but simply by transducing an N-terminal cerebellum receptor, a cerebellum response, the cerebellum binding into an intercellular regulatory response that then controls AMPA and NMDA receptors. And this is schematically illustrated here, where you can see the 2 different neurexins as they bind to cerebellum 2, neurexin 3 and neurexin 1. They're binding to the same transduction machinery, then differentially regulates either AMPA receptors or NMDA receptors. And you might ask, how is it possible that this can work, because after all, this transduction module in the middle is identical for neurexin 1 and neurexin 3. We don't have a conclusive answer, but we believe that this is mediated by differences in affinities that neurexin 1 and neurexin 3 have for cerebellin 2. And thereby, they basically regulate the density of the responses on this GLUD that then differentially transduces one or the other signal. This is just one function of neurexin signaling. I've told you just one particular project, one particular story. Let me tell you, this is important, trust me, because NMDA and AMPA receptor responses are the two flavors of excitatory synaptic transmission. They have completely different downstream effects. They are operated in all new synaptic transmission, not just in plasticity, every time. And their relative activity determines the properties of many synapses and thus of circuits. However, neurexins have many other functions that I haven't had time to discuss with you. And the way I think about neurexins is as a Swiss Army knife, as a platform that does many different things. It recruits calcium channels presynaptically. It does all kinds of presynaptic regulations. It does postsynaptic AMPA and NMDA receptors. It actually regulates LTP as well, GABA receptors, endocannabinoid synthesis, what have you. It's all there. Various neurexin isoforms specify synapse properties via competing ligand interactions. They are the master regulators of synapse properties. So why should you care? Why is this important? If you are not interested in how circuits compute information, which you may very well not be, but you are interested in disease, let me tell you that this is actually very important for understanding both neurodevelopmental and neurodegenerative disorders. This really determines cognitive properties of a brain. And I will give you one example. What you see here is a map of the genetic landscape of schizophrenia taken from a recent paper, as you can see here. And what you can see is that there are some mutations, there are some genetic polymorphisms that are extremely abundant, very frequent, but have almost no effects, shown at the right lower side. And you can see on the left upper side that there are some mutations actually, which have huge effects, but are insanely rare, very, very rare. Importantly, neurexin 1 deletions are probably one of the most, if not the most frequent single gene deletions in many neurodevelopmental disorders, including schizophrenia. It's relatively frequent, much more than the more rare ones on the left, and its effect size is huge compared to the more common polymorphisms on the right. And what you can also see is that this relates to both a role for AMPA receptors and for NMDA receptors, suggesting that there's a pathway in schizophrenia, a genetic pathway, that may very well be a biological one, which links neurexins to schizophrenia. And so with this, I would like to close. What I've tried to illustrate to you is just one project. It's a project that sort of exemplifies how we are trying to solve the issue, the big question of how synapses are actually made and specified. The work we've performed and that I told you about today was largely carried out by a single postdoctoral fellow, Ginia Dai, who now has her own lab at Mount Sinai in New York City. It was also helped by Justin Trotter, a senior postdoc in the lab, and by Jason Aoto, a previous postdoc who now has his lab at the University of Colorado. Thank you very much for listening. I'd be happy to address any questions that you might have.
Thank you, Prof Südhof. We invite Prof Lim to moderate the Q&A.
Well, good morning, ladies and gentlemen, and good evening, Tom. I'm truly delighted to see you today, Tom. It has been a couple of years since we last met during your talk in 2019 at NUS, which I also had the pleasure of moderating. Thank you for joining us here today. Tom, and thank you for once again, as always, your very splendid lecture. I already see participants queuing up, so maybe we could start with the physical attendees first. Please go ahead.
Thank you, Professor, for this insightful talk. So I am Neelkant Kundu. So I'm from India and working in Hong Kong. So I have an engineering background and I have a general question for you. So as a biologist, what are your thoughts on the recent advances in artificial intelligence and artificial neural networks? So will it be ever possible that these AI models can beat this natural human brain, and what are the unique characteristics of brain which may or may not be simulated by a quantum or like a computer? Thank you.
What a great question. Very far away from my area of expertise. However, from my experiences using machine learning and AI in various contexts, including in the drug industry, my impression is that this incredibly powerful approach is better than any human will ever be at identifying patterns, processing big data, sifting through vast amounts of information. However, it is also limited because you have to train the machine in order to do what you want it to do. It doesn't generate its own ideas. It can randomly generate ideas and test them, but the parameter space is infinite. And at some point, that won't work. Let me give you an example. Probably in my own field, the most spectacular success of AI has been AlphaFold, which enables prediction of protein structures. And it's really a major achievement. It is truly something that is very impressive. You can put in any sequence and it will spit out a structure. And that structure turns out to be right many times. But it also has limitations. It can only do that because there's already so many structures. It cannot predict structures which are not conforming to existing structures in some way or another. So if you have something even as simple as something as a new kind of fold, it doesn't work anymore. Moreover, it can only approximate it. Its resolution is too low, for example, to really predict high-level side chain interactions reliably. So in other words, yes, it is fantastic. AI is gonna change how we do science, but it's not gonna change innovation. It's not gonna create innovation. It's gonna potentially make it easier for us in science to discern patterns, to identify commonalities, to make conclusions from vast amounts of data. And how we make these conclusions, what we do with these data, will be very, very important. Let me give you another example, which actually is very important because that's something that many drug companies have learned the hard way. AI is only as good as the database that you use to feed it. When you, for example, analyze RNA-seq data using Go analysis, you get all these functional networks and so on that people show in papers. Most of the time they're wrong. And they're wrong because the data that are fed into these Go analyses are literature data that are not curated, and most stuff in the literature turns out to be not very reliable. And as a result, all these data, all these Go analyses are often, they have some right hits, but mostly they just basically misunderstand. So it's very important here too, that AI will only be good for things where the data are good. It will be great for face recognition. It will be great for pattern recognition. It will be great for RNA-seq data in terms of getting the searching for sequences. But to identify biology, I think its value will be very, very limited. It would be great for drug design, for basically designing potential molecules that bind to drug targets. It already revolutionizing drug discovery. Those kinds of things, yeah, that for sure. But not for, let's say, innovation. I don't think it will work very well.
Thank you, Tom. There is a line of participants waiting to ask you some question. So let me alternate to this gentleman before I take on the online question, please.
Okay. Thanks, Professor, for the talk. My name is Siu Jian Jin, and I'm from Malaysia, working as a postdoc in Taiwan. So my question, I have two questions. The question one is that, is the expressions of neurosin affect the cell-cell interaction between the neuron and other glia cells, such as microglia that actively involved in the synaptic promoting? And my second question is that, do you think the human brain organoid is a good model to study neurosin? Let's say the postdoc in your lab want to use this model. What kind of evidence that he has to show you to convince you it is a good model?
Yeah, these again are great questions. I don't know how synaptic pruning works. I'm kind of waiting for more data on microglia. It's clear that microglia are important in synapse elimination sometimes, but I'm not sure that they are actually generally involved, for example, in developmental pruning. I have my doubts about that. So I know this is by now in textbooks, but I don't think the evidence is very strong. As regards to, what was the second question again? Sorry.
About using the human brain organoid?
Oh, those brain organoids. Brain organoids are really a fantastic innovation to study early development. In terms of circuits, not so great. Their maturity is less than those in 2D cultures. So the 3D cultures, brain organoids, they're never the synapses, synaptic connections. There is very, very little. The best that can be done is by transplanting them. My colleague, Sergio Pasca, has published a recent Nature paper, which I thought was really terrific, that showed that you can get some degree of integration of transplanted organoids, and that may work potentially to study that, but not in isolated culture. You see, the problem is that the organoids, as they exist now, lack basically a blood supply. So, obviously, so as a result, they don't really grow beyond a certain point. They start necroticizing and become funky, and there's lots and lots of problems. And so you can make two different cultures and you can show that one culture extends axons to the other one, but that's not a circuit. That's just basically trying to get around the problem of the fact that it's 3D, but it lacks vital components that the brain has. The brain is not just neurons and glia. It's neurons and glia and the blood supply. And the blood supply is huge in the brain. So I'm doubtful that for studying neural circuits, the organoids themselves are going to be a good model. I'm hopeful that organoids that are transplanted may become a good model, but even there, we're just in the beginning of the field, but that's really where I think the development has to go.
Yeah, thank you.
Okay, there's a flurry of interesting questions online, so let me take one first before circling back to the physical attendee, pardon me. So Tom, there's a question on, is there a potential that your research can eventually contribute to improving the plasticity of the brain for individuals with advanced age? Over to you.
I hope so.
Me too.
So just to get into this a little more detail, we, among others, are trying to identify synaptogenic factors that might be decreasing with aging, with the hope that that will enable a potential treatment in future whereby not just aging, but especially dementia can be at least partly alleviated by increasing synapse numbers, synaptic function as a way of enhancing cognitive performance. I don't know if that's going to work, but it is remarkable that in neurodegenerative diseases, synapses are being lost, and that may be the basis, at least it correlates the best with dementia. And there, if one could prevent that or slow this down, that would be better than an antibody to A beta, at least as far as we know now.
Great. Yeah, I, for one, am really hoping that I could age healthily, particularly when I'm a neuroscientist myself. So let's get back to the online participant. The physical attendee, please.
Hello. Thanks a lot for the talk. I just had a question about the locality of some of these results that you showed us. And in particular, I was wondering, say you have a postsynaptic neuron with two different types of presynaptic neurons that express different neurexins or different types of neurexins. Do they each see some kind of average response postsynaptically, or is the effect localized to the individual synapses?
It's a great question. We have tested 3 brain regions for this paradigm, 3 different synapses. We have not tested the question you asked, which is different synapses from different brain regions on the same postsynaptic cell, but my expectation would be that it's specific for each synapse because it's dependent on the interaction. And so that if you have different presynaptic neurexin variants, you have different postsynaptic responses. In the 3 different brain regions that we have tested, it was very interesting in that in the subiculum, that's what I showed you, the data. In the prefrontal cortex, which we just published in eLife in a very extensive paper a couple of months ago, we see only the NMDA receptor responses. We do not see the neurexin 3 AMPA receptor responses. I have no idea why, because neurexin 3 is there. But that's what we see, okay? So the NMDA receptor response is very strong, AMPA receptor responses, neurexin 3 is not. In the cerebellum, we see only the NMDA AMPA receptor responses, not parallel fiber synapses, not the NMDA receptor responses. That is easier to understand because the parallel fibers don't have any NMDA receptor responses. They lack NMDA receptors, so that's better to understand. And we see a very strong AMPA receptor response. That's where it stands. My hypothesis is that depending on the complement of pre- and postsynaptic ligands, GLUD1 or 2 has to be postsynaptic, neurexin 1 and 3, SS4 plus has to be presynaptic, we will have different responses. In addition, cerebellum needs to be there. However, what makes things a bit more complicated is that neurexins also have other ligands. And these other ligands could compete with the cerebellums. And in fact, we have already shown that in a different paper where we showed that that competition can be helpful in avoiding self-inhibition in the olfactory bulb, which is a study I haven't discussed today. So you really have a very complex system. This is not a simple system where multiple molecules constantly interact with each other, and depending on what concentrations, what splice variants there are, they will form predominant synapses, predominant complexes that then shape a given synapse.
Thank you.
Thank you. I really don't want to keep the 2 ladies standing here for too long. So perhaps why don't both of you ask the question to cut down a little bit of time, and I see whether if Tom could take both the question. Please, go ahead.
Hi, Professor. That was a lovely talk. So I'm currently a PhD student in Cambridge. So my background's immunology, so I don't know very much about neuroscience, but I was just curious as to whether you had anything to say about, for example, Parkinson's and Alzheimer's and dementia, these diseases, they have very different mechanisms of like onset and progression, and it's not entirely clear as to why that's the case, or whether there's a difference in how like neural circuits are degraded specific to these diseases. So is there anything known about what neurexins do in these like specific contexts, or is that still something that's an area of research?
Thank you. Please hold for a while, Tom. Let me invite the other lady to ask the question.
Yes, thank you, Professor. My question is, I'm very curious about this perennial debate between nature and nurture. Because some people say that as humans have a built-in template inside our brain when we're born, and it helps us to build predictive models of the world. I want to ask you as a brain scientist, what might be your insight to this question, or if you have a clear answer to this?
Okay, Tom, two rather generic questions, one on Parkinson's, Alzheimer's, the other nature versus nurture. Go ahead.
So Parkinson's and Alzheimer's disease are devastating and fascinating disorders that nobody understands at this point. There's tremendous genetic evidence which is beautiful and has advanced the field enormously, but for many of the genes, we actually don't know the functions and we don't know how these diseases develop. There's been beautiful studies performed by others that show that in at least some patients, there are changes in the synaptic machinery, including orexins, but whether they're causally involved seems unlikely. I think it's more as an execution of a disease process wherein they might be contributing. We are actually very interested in Alzheimer's disease in particular, and there are talks online that you can see where I've talked about our work there. So this is one of the reasons why I didn't talk about it here, because I thought you could easily access those talks online. And I can tell you that synapses are a key element of these diseases that we need to address, I think, in the field. But at this point, we really don't understand these diseases. And so stay tuned, hope for the best. Hopefully we can, the field, not we, many people, a lot of people who are really, really good scientists are trying to resolve these questions now. And I'm sure that some progress will be made. As regards the nature-nurture question, that is, again, is a very, very important question. There clearly is no yes or no answer in the sense that neither one of them alone explains anything, but both together operate in tandem. I think there's no doubt that experience shapes the brain. And there's no doubt that the environment, in particular the history of a person, has a tremendous influence on brain development and on brain function throughout life. For example, we know from work of others that cerebellums, which I talked about today, are very plastically expressed. Experience causes a dramatic change in the expression of these molecules of cerebellums, not of neurexins, not of GLUD probably, but certainly of cerebellum, suggesting that, for example, at least in some neurons, experience could shape the complement of NMDA and AMPA receptors at synapses. So I think that in this eternal debate about nature versus nurture, we need to resist the temptation to want to find a conclusive one-liner. It's all nature, it's all nurture. Instead, we need to see this as a collaboration between the genome and the environment that together create the outcome, which can be and should be, and hope, a fully functioning, beautiful brain.
Yeah, thanks a lot. There are a couple of more interesting questions online, including one that talks about whether synapses are the driver or otherwise perhaps representing compensatory mechanism for neurodegeneration. Unfortunately, the event organizer has told me that we absolutely has to stop right here now. So Tom, thank you very much once again for the splendid lecture and for your insightful answers to the queries. Ladies and gentlemen, please join me to thank Professor Thomas Südhof for the talk.
Thank you. Thank you.
Thank you very much. My pleasure to actually moderate this session. Have a good day, and to all the Chinese out here, Happy Lunar New Year.
Thank you, Prof Südhof and Prof Lim, for the discussion. We'll now proceed with the next plenary lecture. So our next speaker is Prof Stuart Parkin. winner of the 2014 Millennium Technology Prize. And the title of his talk would be Chiral Spin Textures on the Racetrack. The Q&A session will be moderated by Prof Lam Ping Koi. Prof Parkin, please.
Oh. Okay, first I check the microphone. Can you hear me? Good? No good? Oh, no, it's good. Okay, so good morning. I'm very pleased to tell you something about the field of Spintronics, which is my specialty. And in particular, I'd like to tell you about some recent work concerning chiral spin textures. So I want to give you a little bit of background about who I am. This you can see in this video. This is a recent video we shot of the laboratories that I have built with my colleagues in Germany since I moved there 5 years ago. Originally, I was born in the UK, moved around when I was young from London, Manchester, Edinburgh, Oxford. I went to college in Cambridge, where I'm an honorary fellow at Trinity College today, with a lot of advice from Richard Friend, who comes to Singapore very often. And then I moved to Paris to work with Denis Jerome on organic superconductors. They had just been discovered when I went there. Finally, I moved to San Jose, California, where I joined IBM Research. This is a view of my house in California. But I got married in 2014 to a German chemist, Claudia Felser. So I moved to Germany, and moving from sunny California, then this is a typical picture of the institute in this part of the year. Difficult to get used to. Nevertheless, I want to give you also a brief introduction. So I'm a director at one of the 84 institutes of the Max Planck Society in Germany. And the Max Planck Society has about 20,000 people altogether. And its mission is to carry out world-leading fundamental research. It's one of the premier research institutes we feel in the world, second most highly cited after Harvard. Now, my actual interest is in doing cutting-edge research, but with a view to technological impact. Can we change the world, have impact on society? I think this makes it more exciting when one is at the nexus of discovery and invention and application. This is where my institute is. It's in a small city called Halle. It's a very old city, about 1 hour from Berlin. So the context of my talk is that 500 years ago, a fantastic technological revolution. Johannes Gutenberg, a German scientist in the city of Mainz, invented the movable type press. And this was the first time you could print books. And this concept then of book printing led to the age, if you like, of the Enlightenment, because knowledge could be spread more widely. Now, this age of analog book printing lasted 500 years, so it was an amazing invention. And 50 years ago, IBM Corporation in San Jose, California, where I lived for 30 years, invented a concept called a magnetic disk drive. And this is shown just a picture of a modern-day magnetic disk drive. It's a disk which is rotated under a recording head, and on the disk is a very thin, 10 nanometer thick magnetic film, which is magnetized, so the magnetic moment, the North Pole, points up or it points down. And the transition, and in this picture you can see an image where we have imaged a series of these magnetic regions with decreasing size. Over the last 50 years, the size of the magnetic region in which we store a single magnetic bit, north is 1, let's say south is down, has decreased in size by 1 billion times in 50 years. I want to tell you, the last 4 orders of magnitude was due to Spintronics and some of my own work. So today, if you look in the cloud, what do you see? You basically see enormous numbers of computing nodes, but also enormous numbers of magnetic disk drives. And it was only 10 years ago that a year's supply of magnetic disk drives could store all the information in the world since the beginning of mankind. Now, of course, in the last 10 years, the amount of digital data is expanding enormously with the billions of devices connected through the internet. But nevertheless, this has created enormous wealth and enormous in the form of new companies like Facebook and Google. So if you like, this is a very nice paper which discusses just 10 to 15 years ago, we started to move from the world of books, analog world of storing data, to the world of digital data, zeros and ones, predominantly, even today, in magnetic disk drives that still store more than 70% of all the digital data today. So I want to introduce to you a device that I invented, and it is based on Spintronics. So you're all familiar with current. It will consist of electrons flowing through a wire, let's say. But the electrons have a spin, a quantum mechanical property. The electron spin takes a value +½ or -½ And IBM considered the work that I did one of its most important inventions in the 100-year history of the corporation, which it celebrated a few years ago. Now, what happens with a spin? You're most familiar with this in magnets. A magnet is derived from the electron spins aligning themselves parallel to one another. Each of them has a tiny magnetic moment. And a very interesting memory based on magnetism is something called magnetic core memory. And this is a very early version, 50 years old. You see these small magnetic rings, and these magnetic rings can be magnetized in either a clockwise direction or in an anti-clockwise direction. This corresponds to a 0 and a 1. And through the middle of each of these rings, you can see these copper wires, these green and orange wires. You have 2 wires crossing through each ring. If you pass a current through the wire, it will generate a self-Amperian magnetic field, and the combination of 2 fields from 2 wires at a cross point gives a big enough field that you can change the orientation of the magnetization from clockwise to anti-clockwise. This is a very early form of memory, and it has a particular advantage that it's radiation hard. And this is used for space applications for a number of years. But back now to the electron spin. If you take a non-magnetic metal like copper and you introduce from a battery a current consisting of equal numbers of spin up and spin down electrons, shown as these blue and red dots, then the current is carried equally. The current is not spin polarized. However, if you take a magnetic material, let's say this blue material, could be iron, it could be cobalt, it could be nickel, you introduce this unpolarized current from a battery, and within a few scattering lengths, as the electrons scatter from the magnetic moments which are oriented in a particular direction, electrons with one spin are scattered more than electrons with the opposite spin. This leads to a current that is innately spin polarized. It now carries spin polarization, and an important property of spin is not the magnetic moment, it's the spin angular momentum. And this angular momentum we could use if we deliver enough of these electrons which are spin polarized to create a torque. In the same way that in the mechanical world you can create torques from angular momentum, we can create spin torques from spin angular momentum. I'm going to come back to that later, but here the point is that scattering gives rise to a spin polarized current. Now let's what happens if we take a sandwich. The red layer has a magnetization pointing to the left, the blue layer to the right, and in between we have a thin layer of copper. Each of these layers is atomically thin, let's say a few atomic layers thick. We can make these materials by simple sputtering techniques, which I'm not going to discuss. If you introduce a current into this material from the battery, then the red layer prefers the red electrons, spin down, and the blue layer, the blue electrons. But however, the copper layer is so thin that the electrons will be moving in all directions, and the blue electrons will be scattered in the upper red layer, and vice versa. This means somewhere in this structure, all the electrons are strongly scattered, high resistance. Now all we have to do is apply a magnetic field, rotate these magnetic layers which had anti-parallel magnetic moments to become parallel. Now the blue electrons can now be short-circuited through this sandwich, and so the resistance is lowered. So basically, by applying a magnetic field, we can change the orientation of these atomically thin magnetic layers, and we change the resistance. This is a magnetoresistance phenomenon. Now this phenomenon of spin polarization of the current was actually proposed in the 1930s by Neville Mott. Neville Mott won the Nobel Prize for an amazing series of discoveries. When I was a student in Cambridge, his office was the opposite the region where I was building and exploring transition metal dichalcogenide magnetic systems. Nevertheless, the change in resistance in the structure is only 10%. Nevertheless, this device I proposed as a sensor to detect magnetic fields, to detect the magnetic regions in a magnetic disk drive, and we could do that 10,000 times more sensitively than any other device that's known even today. This is what enabled us to increase the storage capacity of magnetic disk drives 10,000 times in a very short period of time. Now there's another very interesting, closely related spintronic structure. This is called the magnetic tunnel junction. So you see again, you see the red and blue layers, that corresponds to their magnetic moment direction. But now instead of a metallic layer in between, we have this thin gray layer. This is an insulator, no electrons. So the electrons now quantum mechanically tunnel from one metallic electrode through the insulator to the opposing metallic electrode. Now the advantage of this system is that the resistance can be much higher for current perpendicular to the layers than in a metallic structure. A metallic structure, a few atomic layers thick, as in the spin valve, there's virtually no resistance in the perpendicular direction. This system has high resistance. Moreover, it has high tunneling magnetoresistance. The reason is when you extract current from the surface of a ferromagnet, there are different numbers of electrons with spin up and spin down. They tunnel at different rates, and when the two magnetic layers are parallel, the electrons can tunnel readily of one spin polarization. You switch the orientation of that one layer to the blue direction, and you essentially turn off the current, because there are no available empty electronic states into which those spin polarized electrons can tunnel, because the spin is preserved during that process. So this type of structure can give rise at room temperature to changes in resistance when you change the orientation of these two layers of up to several 100%. And this is what we demonstrated in this paper in 2004, and is now the basis of a new technology to store information in what is called magnetic random access memory. And just like the cross-point array for the little core memories I showed you earlier, we have a very similar idea, a cross-point of wires at each intersection. We introduce a magnetic tunneling junction device where the current flows from the upper wire to the lower wire through the device. And this tunneling junction has sufficient resistance that we can detect its resistance, whether the moments are parallel or anti-parallel, a 0 and a 1 in a tiny amount of time in less than 1 nanosecond. And at the same time, if you look at the top, you can see this, remind you again, this is the magnetic disk drive. We incorporated the metallic spin valve into this recording device, which moves mechanically over the top of the surface of this magnetic layer at a distance today of about a few nanometers, enabling us to read the transitions between these magnetic blue and red regions with greater sensitivity than was ever possible before. Turns out this metallic spin valve was used in magnetic disk drives for 10 years, 1997 to 2007, was then replaced by this magnetic tunneling junction using the materials that we proposed in 2004. Now, just as a comment, I wanted to point out this. We proposed this concept of a magnetic random access memory, which is a non-volatile, we've demonstrated high performance memory that can be scaled to smaller dimensions than charge-based memories, but it took from our proposal in 1995. We first demonstrated this in 1999. We had a 16 megabit chip we demonstrated in 2005, but it took until 2019 that Samsung announced you can now buy in their foundries this embedded magnetic random access memory. And typically, it takes 20 years or more from the discovery of a material to its application. On the other hand, surprisingly, the spin valve sensor, we could, using the physics I discovered in 1988 to 1991, IBM could incorporate that into magnetic disk drives in just 6 years, which is remarkable, allowing, as I said, a 10,000-fold improvement in the capacity of magnetic disk drives. So I want to now discuss briefly another memory that I'm even more excited about, and I actually also proposed this in 2002. So now it's 20 years later, and not yet available, but we believe, or I believe, it's on a path to application. And the concept is entirely different from any other memory technology today. You see in this cartoon, you can see a magnetic wire. This represents a magnetic wire, which is nanoscopic in size and maybe a few hundred nanometers tall. And this wire is a uniformly magnetized magnetic heterostructure. The red and blue regions magnetized in one direction and the opposite direction. And in between these regions, you have a so-called magnetic domain wall, which on a nanoscopic scale looks like a particle. It has mass, it has momentum. And what I proposed was we could pass current pulses into this wire. It becomes spin polarized and delivers spin angular momentum and can move these domain walls synchronously around the wire. You move the stored information to individual reading and writing elements we would build into the racetrack. That's what I proposed in 2002, and it took till 2008, and I'm very proud of this paper in Science. It's nearly 5,000 citations, and this is a fantastic achievement, I think, where we demonstrated this basic principle. But again, we had to go beyond that and discover much new physics to make it relevant technologically. How does this work? As I mentioned before, you take a current in a magnetic material, it's spin polarized. And that spin angular momentum of these conduction electrons shown as these moving electrons in this cartoon, we can transfer that spin angular momentum to the magnetic moments in the domain wall. Roughly speaking, every time we introduce a polarized spin, it can rotate a single magnetic moment. So we need to pass enough current through this material to move the domain walls. And that's something I said we demonstrated for the first time in 2008. Due to the short time, I can't tell you all the very interesting physics, but I like to consider that this concept of a racetrack memory has gone through 4 important stages. And I want to briefly discuss some of the physics in the final stage, 4.0, because that is the racetrack that is being considered today as a possible replacement for magnetic disk drives. It would have the same storage capacity, but would be 1,000,000 times faster and use 50 to 100 times less energy and would be totally reliable. Magnetic disk drives, because they're mechanical, can fail. So there are 2 things I want to discuss, both of which are chiral. First of all, many of you are familiar with the Hall effect. If you pass a current through a non-magnetic material, in the presence of a magnetic field, the electrons will suffer from a Lorentz force, and they will move either to the left or to the right. So you basically create a transverse voltage, which is not spin polarized. Now there's an equivalent effect to the Hall effect called a spin Hall effect. You don't need any magnetic field, but in certain metals where the spin is coupled to the lattice through what is called a spin-orbit coupling, you can create a transverse spin current. And this is a chiral phenomenon. So if on the right top, you can see this material, this pink material, it turns out, let's say it's platinum. We pass this current yellow through the material, and you can see these arrows, they correspond to spin polarized electrons that accumulate on all the surfaces in a chiral direction. When you change the direction of the current, the chirality changes. So on the surface, we'll end up with accumulation of spin polarized electrons, no charge accumulation. And now what happens, we can use that spin polarized current and pass it into a neighboring magnetic material to create spin angular torques. And that is what we demonstrated. And today the efficiency of converting one electron into a spin polarized electron exceeds 1. Some recent materials we published earlier last year, we demonstrated that every charged electron can generate 2 or 3 spin polarized electrons. That's the first. This is a chiral phenomenon. The chirality of this spin current depends on the material. Another chiral phenomenon is the domain wall itself. So going back to this very simple picture, you can see here is the racetrack, magnetization here pointing in the red direction up, blue direction down, but in between the magnetization rotates in a very special way. It will rotate in either a clockwise or an anti-clockwise direction. You can see at the bottom, you can see here is the magnetization pointing down, and as it rotates to the up direction, you see in the middle, the magnetic moment here is pointing to the left, but when it rotates from an up to a down direction, that magnetic moment points to the right, and it turns out it has a chirality. This is introduced by the same material in which we see this spin Hall effect. We grow our regular materials. The racetrack is typically made from, let's say, cobalt, nickel, cobalt. These layers are 1 or 2 to 3 angstroms thick. And underneath, we have this orange layer, which could be platinum. Platinum not only gives the spin Hall effect and spin currents, at the interface, it gives rise to a very interesting type of proximity exchange, which is called a Dzyaloshinski-Meria vector exchange, which gives rise to these chiral structures. Very important. And another point we have to take advantage of or take control are dipole fields. So you all know that a magnet generates fields. So the Earth has its own magnetic field. Now, these magnetic fields are long range and cause interactions from one racetrack to the other on the nanoscopic scale. They increase approximately inversely with the size. So even for the spin valve device, originally one had to control the dipole fields from one of those magnetic layers in the sandwich. And I had proposed, even at that time, we could do it with something called synthetic antiferromagnet. And this is the only reason why we could make spin valves and why we can make magnetic tunnel junction MRAM devices. So if you look at this cartoon, you see here is a lower racetrack where the magnetization rotates from up to down. with a chiral, this is actually a nail wall because the moment here is pointing perpendicular to the transition. We create on top a second racetrack, which is exactly a mirror image of the lower one. So the moment in the lower racetrack is exactly mirror imaged by an opposite moment in the upper racetrack. In this way, all along the racetrack, the net magnetization is 0, and we can eliminate the dipole fields. Without this, we cannot build any type of magnetic device. And this actually was an invention of mine based on my discovery of long-range oscillatory interlayer coupling. It turns out if you take a thin layer of ruthenium, 7 angstroms thick, it will give rise to this amazing strong anti-ferromagnetic coupling, so we can make this mirror copy. By making this mirror copy, we discovered if we now pass current into this racetrack, the velocity of the domain walls at the same current density as we make it more and more balanced increases by up to a factor of 5 to 10. So now we can move these domain walls 5 to 10 times faster than we could have imagined when I first started this project in 2002. This is totally new physics, but it means we can move these domain walls extremely efficiently at high speeds, exceeding 1 kilometer per second, in these racetracks where there's no net magnetization, no dipole fields. Perfect, the only way to make an application. Now, how do we see these domain walls? So when I was at IBM, we did this by microscopy, so-called Kerr microscopy. You shine a light which is polarized in one direction, and when it's reflected from a magnetic material, the plane of polarization slightly rotates to the right or to the left, depending upon the direction of the magnetic moment in this racetrack. We take a snapshot, and then we apply a current pulse, and then we take another snapshot, and we can see then 20 domain walls you see here moving backwards and forwards in a regular racetrack, a single ferromagnetic layer. Underneath, here's a cartoon of this synthetic anti-ferromagnet. We have this platinum layer, we have these 1 nanometer thick cobalt, nickel cobalt, ruthenium, 7 angstroms thick, cobalt, nickel, cobalt, and this has no net magnetization, so it's more difficult to image. These domain walls here are moving much faster. So this is because of chirality of the domain walls. If we could not control the chirality, the domain walls would be moving in random directions. They only move in the same direction with current if they have the same chirality, and it's based on spin currents derived from this spin Hall effect. This is what makes it possible. Now, at Max Planck, since I came, we have, with some funding from Samsung, we've shown we can go from micron-sized racetracks down to nanometer-sized racetracks. Turns out everything works exactly the same velocities, and we've now built, for example, a device where we have a single domain wall. We move it backwards and forwards and detect its position from a integrated magnetic tunnel junction, and we can do this on a time scale of less than 500 picoseconds and extremely low energy. So we believe this is a very interesting single domain wall device, super high performance, which we could use to replace the densest memory today in charge-based world of electronics, which is called static random access memory. Now, probably running out of time, but I want to briefly discuss, if there's time, we can go beyond domain walls. And it turns out 10 years ago, Christian Fleidrer at the Technical University in Munich discovered something called a block skyrmion, a magnetic material where normally the magnetization is fixed in a single direction. He discovered that as a function of temperature and magnetic field, suddenly it forms an hexagonal array of tiny objects called skyrmions, whose energy is lowered by the formation of a chiral domain wall using exactly the same methods that I briefly discussed earlier. Now, the earlier domain wall, you can also form these skyrmions with a nail domain wall, similar to the domain wall I showed you in our racetrack. In this case, the magnetization rotates. Outside is pointing up. Inside this object, it's pointing in the opposite direction. And along this radius, the magnetization rotates in a chiral direction. In the block skyrmion case, the magnetization rotates not along the radius, but perpendicular to the radius along the tangent. And it turns out that with theoretical predictions, we could see another object called an anti-skyrmion, where in this case, because of the symmetry of the crystal lattice, this Dzyaloshinski-Maria vector exchange has different sign of its vector components as you move around in the lattice structure, if you have the right symmetry. And there was a prediction you could see something called an anti-skyrmion, where the boundary alternates between block-like domain walls to nail-type domain walls. And we were the first to discover this. And how did we do that? We discovered it by taking a single crystal, Turns out these were made by Claudia, my wife, in Dresden. And we take a very thin layer. We cut a thin layer out using focused ion beam milling techniques, which is electron transparent, less than 200 nanometers thick. We pass this high energy beam, 300,000 volt electron beam, through that layer, and we detect through Lorentz forces, the same Lorentz forces that give rise to the ordinary Hall effect, the electrons are moved because of forces from in-plane components of the magnetization. And a nail skyrmion, actually, if you image it, you'll see nothing, but a block skyrmion, you'll see the electron beam is focused towards the middle of that skyrmion. It would look like this, and we predicted that an anti-skyrmion would show, as the electron beam was transmitted, there'll be two regions of high intensity, white, two regions of low intensity, black, and I think you can see, here is the image we saw. This is an anti-skyrmion. The first observation, it's about 200 nanometers in size, and we published this 5 years ago, and we showed that as a function of magnetic field and temperature, we could evolve these structures into more complex beasts. And also we found that by simply changing the thickness of this layer through which we image these anti-skyrmions, you can see this, in this case, this is an image using a different technique. We basically take a magnetic tip, move it across the surface, look at the displacement, and we're now looking at the outer plane component, and these little dots correspond to anti-skyrmions increasing in size as we increase the thickness of this layer through which we're looking from a few 100 nanometers to 4 microns. Now, this whole area of science of these non-collinear spin textures has been very interesting in the last few years, and we ourselves showed the same material in which we can see these anti-skyrmions. We actually can see another object. You see these elliptical objects where we can stabilize these objects because the wall now is entirely of a block wall type, which has the lowest dipole-dipole energy interaction. So we can convert anti-skyrmions into elliptical block skyrmions, and one proposal we have is maybe we could build a racetrack where we store information not as the absence or presence of a domain wall, but as an anti-skyrmion or an elliptical block skyrmion. This will be very difficult to do, but we can see a combination of these objects experimentally, and of course we can simulate this using micromagnetic simulations. Now, how much time do I have? 1 minute? More time, okay, I have more time. So I want to finally talk about our current project, which is, I propose this, only been working on this 2 years, and we already think it's super exciting. We want to make what I call a super track, a racetrack memory that would operate at ultra low temperatures to support quantum computing. So many of you are familiar with quantum computing, a lot of excitement, and one of the most exciting quantum computing ideas is to use a spin as a qubit. I'm not going to talk about that. But if you ever want to make this happen, it will operate at very low temperatures, and you need a memory, a conventional memory, that will store the parameters to set up the quantum computer and to read the output of the quantum computer after it has performed its function. To do this, you need a memory that operates at ultra-low temperatures, 10 millikelvin. And basically today there is nothing. However, racetrack comes to the rescue, and with racetrack we can build a super track. And we want to do this using two ideas. One is we want to build supercurrents in which the supercurrent is carried by so-called triplet pairing. So perhaps many of you know in a superconducting material, again, the electrons pair up with opposite spins to create a boson, and you form a macroscopic quantum state in which the bosons can be moved with no dissipation. So basically no resistance, no voltage. Well, this is called a supercurrent. Now, it turns out that if we could create that same Cooper pair with the electron spins parallel, in principle, this is called a triplet pairing, we could create a supercurrent carrying spin angular momentum. Therefore, we would have a current without any dissipation, which we could use to manipulate magnetization. This has never been demonstrated, but 2 years ago we demonstrated that by, again, coming back to these non-collinear spin textures, we could use one of these to create, by proximity with a conventional superconductor, this is niobium, where the Cooper pairs are formed from anti-parallel spin pairs, we could create triplet pairing in this non-collinear spin texture. Don't have any detailed time to discuss this, but this is a chiral Kagome antiferromagnet, and we showed the supercurrent could be propagated over long distances. And more recently, demonstrated something for the first time, which probably I don't have any time, it's called a Josephson-Diode effect. We've demonstrated, now you can imagine, if you pass a supercurrent in a material, if the current is too big, eventually you destroy the superconductivity. And there's a supercurrent critical density below which the supercurrent will flow with no dissipation, and above which it looks like it becomes dissipative. You destroy the Cooper pairs. Well, it turns out normally, you can see in this picture, we've taken this, you see this little irregularly shaped, it turns out it's a 2D material. It's a nickel ditelluride. It's a very special material, which is a Dirac semimetal. It has surface states which are spin polarized. And then on top of this, we create these wires. These are formed from niobium, a conventional S-wave superconductor. We pass current into this non-superconducting material. It becomes superconducting by proximity. And we found at these lowest temperatures that the critical supercurrent density for current flowing left to right or right to left was the same, but in the presence of a tiny magnetic field perpendicular to the supercurrent, we found that these critical supercurrent density is distinctly different. First time, Josephson-Diode effect. And this means that if you pass a current to the left, it's dissipation free. You pass it to the right, it will be dissipative. So we could use this to detect domain walls. So my current program is we want to build a super mint or super track, a cryogenic racetrack memory, which would basically consume very little energy. We would create triplet supercurrents in the very special ways, pass them into the racetrack. They would deliver angular momentum, the domain walls would move, and we would detect them using this Josephson-Diode effect. So we think this is a very interesting concept, and we're working hard on this. Final comment, in the 5 years I've been at Max Planck, we built this system. We believe it's one of the world's most sophisticated systems for building atomically engineered materials using different techniques that are required to deposit different types of materials. So we have molecular beam epitaxy here, but this is something we built ourselves. We call it Mango. It has 44 different sources where we can sputter using magnetron and ion beam sputtering. We also have something called pulsed laser deposition, and all of these systems are connected through this. You see this orange tube, it's 15 meters long. And we've designed it so we can automatically, under computer control, take a substrate from one of these systems to another system and deposit layers from one atomic layer to several atomic layers thick to create entirely new materials that we hope will change the world. So, summary, spin valve sensors, they're green, basically makes possible the digital world of today, I like to think. Magnetic tunnel junctions, it's green. We succeeded, it took 25 years. High performance, non-volatile memory. It's one of the most interesting emerging memories today. Racetrack memory, you see a little less green. We think with the materials we have and the new physics we've discovered, this could replace magnetic disk drives in the next 5 to 10 years, 1 million times faster, 100 times lower energy, and even we could replace the fastest charge-based volatile memory today with a non-volatile memory, will be smaller in size, use much less energy, and would make computing systems much more powerful. And of course, the whole zoology of non-collinear spin texture is super exciting, not clear they would have any real application. And finally, will be an application, triplet superconductivity. By the way, there is no known bulk triplet superconductor, but we can create it using superconducting spintronics. And I think this cryogenic racetrack, again, could be a fantastically interesting memory that could support quantum computing. And if you're interested, here's some of our most recent papers on these two concepts, racetrack and this supertrack. And then I just want to acknowledge all of my collaborators, my students and postdocs in Halle. We collaborate with other groups around the world, including Liang Fu's group at MIT and Takis Kontos' group at the École Normale Supérieure as well. Thank you very much for your attention.
Thank you, Prof. Parkin. We invite Prof. Lam to moderate the Q&A.
Okay.
Thank you, Professor Parkin, for this really impactful and exciting talk.
Okay, thank you very much.
Yeah. So we can now take some questions for the online attendees. Let's start with toggling between the right and the left.
Oh, wow.
Hi, Stuart Parkin. Hi, Professor. I'm Timothy from NTU, Singapore. So Singapore is very excited about artificial intelligence, right? So we know that AI requires large computational models and large neural networks. that consumes a lot of computational power and memory storage. So what do you think about your new, your magnetics, your racetrack memory, right? How can it impact how we do AI in the future?
So AI is basically made possible by the fact that we can have so much data stored digitally, which is the lifeblood of artificial intelligence and machine learning. You need to store that, all that data has to be stored. And of course, magnetic disk drives consume a lot of energy. So today, currently, about 7% of all electricity is consumed by data centers where the data is stored. So if we could reduce that energy by 100 times using racetrack, that would have a huge impact. We'd eliminate 7% of all the electricity used today. So that's one way in which I think racetrack could play a very important role.
Thank you.
Okay. Thank you for your interesting talk. I have a basic question about the data security in computing. Like, what could we do to keep the security in data production and transformation or use of the data, or also like writing or reading the data in storage?
Well, you know, that's a very interesting question. Little to do with Spintronics, but nevertheless, it's a super interesting question. Like, even Max Planck, I know a major institute suffered a ransomware attack just a few weeks ago, lost all their data. This is a super important problem, cybersecurity. And it's not clear to me that, well, how Spintronics could address that question. It's a very interesting one. I don't really have an answer. But make sure all your data is backed up beyond a single magnetic disk drive.
I will. Thank you.
Next question, please.
Hi, Professor. Thanks for the wonderful talk. So there's a new emerging field of optical skyrmions, where you tune the local polarization states of light to create these non-trivial topological states. Mm-hmm. And in the material science world, people use them, and they normally quote in their papers that they are resilient to noise and that to perturbations. What kind of perturbations are these? Because whenever we try to publish papers on optical skyrmions, we get this criticism that it's not unclear what kind of noise or global operations that these things are invariant to.
Okay, so I would answer two ways. One is, of course, optics, very, very interesting, but and the concept of using different types of optical phenomena to store data is a very, very old one. However, typically, photons, simplistically, are too big. So it's difficult to make devices where you can store enormous amounts of data using photons, unless you use super resolution techniques, for example. Nevertheless, I would say more interesting in my mind for skyrmions is the use of optical probes to manipulate the skyrmions, to change their structure, to create them by using the photons themselves, intense photon beams. And this has been demonstrated that, for example, you can reverse magnetization or you can create regions which are skyrmions using intense optical probes. I think this is much more interesting, using ultra super resolution techniques.
And what noise mechanisms are they resilient to?
So this concept, you know, optical beams can carry angular momentum, and angular momentum again can be used to manipulate magnetization, but it turns out at the same time you get a lot of heating. So this has been very controversial. It was shown in the 1990s that you could reverse magnetization of a magnetic material on a picosecond timescale, which is why it's interesting, but nevertheless, it was mostly determined to be by heat. So thermal effects play a very important role.
Thanks, Prof.
Questions from here.
Hello, Professor. My name is Shopnadeep. I'm from India, and I completed my PhD from Hong Kong. So my question is for the emerging memory technologies. For example, if we compare the magnetic RAMs with the phase change RAMs or the resistive RAMs, what gives MRAM its competitive edge over the other types of emerging non-volatile memories, particularly for speed and device scalability, when our RAMs have already reached like 4 nanometer square size and speed of 85 picosecond? Is it the device-to-device variability, or what factors make MRAM more commercially viable compared to?
So our RAM is very interesting, and there are different approaches to building resistive random access memory, including phase change. And then this memory has been under development for 30 years and has not found its way into the marketplace, or it did briefly. And the reason is that when you, let's say, a phase change memory, which you change a material from a crystalline to amorphous state by going to high temperatures and changing the cooling rate so it cools into an amorphous state or cools more slowly into a crystalline state, it turns out you get thermal expansion or contraction, and this basically causes damage. And it's not possible to do that reliably over long periods of time. So typically, any memory has to nominally survive for 10 years after trillions of operations. The phase change resistive memory concepts don't appear to be very reliable. So there have been no resistive or memory or phase change memory has made its way into the marketplace for these reasons. So I would say magnetic materials, on the other hand, where we only, let's say in racetrack, we don't move any atoms. Anytime you move atoms, you typically have problems. So similarly, ferroelectric memories, been around for a long time. Again, you have to move atoms. These atomic motions eventually cause failure. However, magnetic racetrack, we only rotate the spin, nothing to do with the atoms. Therefore, super reliable.
And how easy it is to tune the different resistance states in MRAM compared to RRAM, PCM, or FRAM for the neuromorphic computing applications?
Yeah. So again, for neuromorphic applications, racetrack memory is fantastic because you can build a racetrack in which you basically move a domain wall in one magnetic layer and not in the other one. And then if it's basically a magnetic tunneling junction, you can create 1,000 analog states, which is typically what you need for neuromorphic computing applications as a resistive element in a cross-point architecture. So this means we can create highly reliable, highly controllable states. And I didn't have time to discuss this bit on our racetrack. We've demonstrated we can move a domain wall with a precision of better than 40 nanometers for positions 100 nanometers apart. So if we can build a vertical racetrack, which we're close to doing, we could build an element that would be, I think, perfect for artificial synapse. So we just heard about synapse, but of course we and others are trying to create artificial synapse using non-conventional, non-biological materials.
And in terms of the scalability of the devices, so those typical RRAMs or RRAM specifically, they only depend, the device size depends on the top electrode size. So isn't the scalability of the devices much easier compared to MRAMs where you have to scale down the magnetic domains and they have a limitation?
Well, another problem with resistive random access memory, which typically relies on moving atoms, let's say oxygen atoms in a material, is that typically you create an atomic connection, and you're moving atoms around within that atomic connection, and it's unreliable, and moreover it's unscalable. Basically the same independent resistance, independent of size. And then the thermal effects become dramatic on the nanoscale, so it does not scale very well. On the other hand, magnetic random access memory has already been scaled to dimensions below 10 nanometers by Samsung, so it seems like it's totally scalable.
Let's take a break from the attendee, and then we will look at one of the online questions. One of the questions online asks, how long do you estimate that a quantum state could be retained in the super racetrack memory?
Oh yeah, it's very interesting. So we just have a proposal, we just put it on the archive with Daniel Loss, who's a theorist at the University of Basel, who's one of the pioneers in understanding quantum computing. And we proposed that we could use one of these chiral domain walls as a qubit, where the 0, if you like, we would change from a clockwise to an anti-clockwise domain wall. And this, we believe, could be a very interesting theoretical proposal for a qubit. This is something we're working on.
Okay, so what sort of time scale would you expect to have those memory lasting?
Oh, timescale. Oh, well, we haven't made any such device yet, but we would, of course, anticipate that we would design it so the timescales would be related to whatever barrier was needed to go from one state to the other, and so it could be whatever time we wanted. Could be long.
All right, thanks. Yep.
I believe. Since we haven't done it, it's easy for me to say.
Next question.
Yeah, thank you very much for your talk. I was really impressed by, in particular, the images. And actually, when you introduced the racetrack, you had this beautiful illustration of the mirror image of the domains, which I couldn't help but think of DNA.
Oh, okay.
And I was wondering, and I'm glad that Mr. Klenerman is in the room, is there a realm where you could use racetrack and the high speeds to sequence DNA, for example, using microfluidics through a channel, and combining it with the unique chirality of each nucleotide?
Well, that's a really interesting question. I think maybe you should answer the question. But let me answer it differently. It turns out there have been interesting ideas to use giant magnetoresistance to separate molecules. by taking nanoparticles. And then on the surface, you put some kind of chemical that will be attracted to some aspect of DNA or something. And you could use this to separate molecules in this way. But I think that's in its infancy. Concepts are now more than 15 years old. So I don't really have a good answer to your question. Very interesting one.
Thank you.
But no answer, sorry.
Thank you, Professor, for the very interesting talk. My question is more on a basic scale. I understand from my work at the Institute of High Performance Computing that different applications have different requirements for data storage. So whether that be speed, reliability, or capacity. So when you design the racetrack memory, how do you balance between all these three requirements? And could you comment on, in your opinion, which of these are the most important when designing memory for high performance computing applications?
Okay, so high-performance computing, you have a whole series of memories at different levels. And so level 0 would be on the chip itself. On the chip, you would build typically SRAM because that's compatible with the transistors for the circuits. However, as you know, half the chip today is SRAM. And it's anticipated as you scale to smaller and smaller dimensions, the size of a single SRAM, single static random access memory, will be expanding. So it's a few hundred so-called F² today, the minimum lithographic dimension, is going to explode. So it's not possible. So if we could use a single domain or racetrack to replace static random access memory, that would already have huge impact on high performance computing, because at the L0 level, we could put in more memory. And it would be non-volatile, therefore would use less energy. But as you go into the stack, into high levels, of course, eventually, if we could replace all the disk drives with racetrack, that would also be a much faster and reliable. It would be 1 million times faster. So that would have huge impact on computing systems. But your question is a very good one. So in some sense, you have to design these memories to have different performances and different densities, let's say. So typically, you have SRAM, big, very not dense, but high performance, less than a nanosecond. But with racetrack, we can get density and speed. So it looks super interesting.
Thank you.
One last question before we break.
Thank you, Professor. My question is about applications to outer space conditions. Like what could be the possible changes to the magnetic properties in space conditions and effects on the magnetic sensors, such as spin valve sensors and skyrmions? Thank you.
So it turns out that when we first proposed magnetic random access memory in 1995, it was because the military in the United States, the so-called Defense Advanced Research Project Agency, wanted a memory that was radiation hard. And it turns out all charge-based memories have problems that you create in space. Radiation creates charges and can destroy the memory. But magnetic random access memory, because the information is encoded in the magnetic moment, has no such issues. So it turns out for space applications, these magnetic memories are very important, because they're not charge-based. And it turns out this core memory I showed you was one of the very earliest memories used in space, because again, it's radiation hard.
Thank you all for contributing to these very interesting questions. I think it's time for us to break for lunch. Please join me again to thank Professor Stuart Parkin.
Okay, thank you very much. Thank you.
Thank you, Professor Parkin and Professor Lam. That concludes this morning's session. I have a few housekeeping announcements. This afternoon's lunchtime online engagement is with the Singapore ETH Centre on Envisioning the City of the Future, which will take place on the Give Me platform. And for the poster session participants who have yet to collect your cert, please collect them at the registration counter. And I also like to bring your attention to 2 items that we found outside during the morning tea break, which is a pink umbrella and a water bottle in a blue sleeve. So if they belong to you, please collect them from me. Yeah, otherwise we'll break for lunch and we'll see you back here at 1:25 p.m. Thank you.